Well, hello everyone. We are live. David F. here. Sal Cordova is running a little bit late. So while we are waiting for him to come back, we do have Jackson Wheat here. So I guess we can just get started a little bit. Jackson, tell us a little bit about yourself and what's going on your channel.
Uh, well, I am an undergraduate currently. Hope about to be a graduate student. So, in organismal biology, and my channel is about evolution and combating young Earth creationism. Although it's predominantly just about teaching evolution, phylogenetics, and those sorts of things.
Okay, very nice. And I am going to find a link to your channel and put it in the side chat. And so my name is David F. I'm also an undergrad. I am getting ready to transfer out of community college into university. It looks like I have a pretty good chance of being accepted to the University of Colorado at Boulder. So that is where I will hopefully be going. I will be doing the molecular cellular developmental biology program, and we'll be doing a minor in the astrophysics and planetary science, hopefully.
Wow.
So yeah.
A lot of stuff.
Yeah, definitely. And a lot of math involved.
I bet. Yeah.
Yep. Hey, Leophilius, I see you in the sidebar chat. Hey, Leo, you should join the stream. Leo, do you know him?
I believe I've heard of him. So, hi, RJ.
Hey, RJ, it's good to see you. We are, of course, sadly running a little bit of a delay on Sal's end. So we will be getting started soon. So this kind of started last night. Myself, Jordan, and Leo had a very good conversation with Sal Cordova on his channel on the topic of radiometric dating and other dating methods. And so it was a really, really lovely conversation. And so this is kind of like a little bit of a spillover. So today we're going to be talking about eukaryotic evolution, the origins of life, and human evolution as well. So lots of stuff to look forward to. Leo, I sent you a link on Twitter. So you should be Leo should be coming in just a few minutes.
I'm afraid I'm not very knowledgeable on the topic of abiogenesis. I'm not a biochem person or an organic chem person. I have had to take organic chemistry, but I was not a fan of it. Organismal biology is my
very nice area. Yeah, so I imagine we'll be talking more about eukaryotic evolution. I'm not too knowledgeable on abiogenesis myself. Organic chemistry is one of the things that goes way over my head.
I mean, it's interesting. You know, some of the experiments are interesting and some of the results, but sort of at arm's reach. Organic chemistry was a lot of take this molecule and turn it into this molecule, you know, and the stereoisomers and all that. And that was not fun. I did not enjoy that.
Yeah, I would not enjoy that either. But I do like chemistry and I do like biology, so and astrophysics. So a lot of stuff I like. Hello, Dr. Dan. I see you in the sidebar chat. Sorry you're not able to join us in the live stream. Again, we're running a little bit late, but that is okay. So we're just going to have a little bit of some coffee while we're waiting.
Hello, Daniel.
So, if you don't mind me asking, where are you in undergrad at?
Louisiana State University of Shreveport.
Oh, very nice.
Yep, it's good. You know, everything goes well. I will have my degree at the end of this semester, which is next Friday. So.
Oh, very nice. Yep, today was my last day of finals for my class. I submitted my written final and got my jury in, so all of that is in. Nice. So now it's just the waiting game. I don't see why I shouldn't have anything less than an A minus. So hopefully I didn't fuck up too badly.
Yeah, I don't think it went too badly. Everything was pretty easy. with the COVID, everything's a Zoom, so they're just like, here's a project, turn it in. Wow.
Yep. So, where do you once you go for grad school? What do you want to get your graduate degree in?
My graduate degree will also be in organismal. I'll still be here at LSUS. Fortunately, they have a grad program, but then for my doctorate, I don't know where I'll go.
Yep.
I haven't thought about it.
Yeah, I kind of wanted. I've always been fascinated by virology and viruses, and especially with the COVID-19 thing, it's kind of really gotten my interest in going into MCDV and virology. And I know Dr. Dan is a virologist also, so um, definitely he's been a pretty big inspiration to me to go that route.
Yeah, well, virology's okay. I prefer more macroscopic animals or organisms.
Yeah, I feel you. Yeah, I feel that virology probably will open more job opportunities with vaccinations and stuff like that, and immunology. I'm also interested in oncology and all that, so I'm not quite sure exactly which direction I'm going to go, but we'll see.
Yeah, exactly. Yeah, I'll probably my plan is to go into zoology for my doctorate to specialize in some group of animals.
Yeah,
I mean, plants are okay too, but I prefer animals.
yeah. Plants just bore me to tears. I'm sorry,
Oh,
but botany is just so not interesting to me.
you see that? I got a phylogenetic tree back there. All those green ones. That's. I also have another one, but it's on the opposite wall. Um, I think plants are neat. There's a lot of really interesting aspects to plant evolution. It seems that the fundamental divide between the ancestors of plants and our ancestors was our ancestors had one flagellum, while ancestors of plants had two.
Let's see here. Dr. Dan says, after a fly lab rotation, never macroscopic ever again. Oh boy, Dr.
Oh, I
Dan.
love arthropods. What are you talking about?
Oh man, I'd love to hear that story, Dr. Dan.
Yeah, I mean, I've heard other people say the same thing, but I don't know. I'd much rather work on animals or birds or mammals. I was in while I was at LSU Baton Rouge. I was in the Department of Mammalogy, and so the professor I was working for specializes in shrews and shrew rats, and so he actually discovered a couple of new species of shrew rats. The most interesting one was there was one that was sort of partly aquatic. It was called Wyomes, and that was kind of neat. But the one that I really thought was cool was Tomis vermidex. It's this shrew rat from Indonesia. Well, they're both from Indonesia, but it has this very long rostrum. And the reason is it sucks up earthworms. That's what the name means. Tomis vermidex, few-toothed shrew rat earthworm destroyer vermidex. And so, but it lost its molars, which is really weird for a rodent because the diagnostic dental pattern for rodents is incisors, diastema, molars, and it's just incisors, no molars. It's really weird, but it's very honed for eating earthworms. So
Interesting.
yeah.
So, we're just waiting for Sal to get back. So, anyway, is your
Uniconta versus Biconta. That's true.
yeah, sure. So, I guess for those who don't know, can you explain the terms Uniconta and Biconta?
Us and our ancestors. So animals, fungi, Philisterians, ichthyosporians, amoebozoans, we're all members of Uniconta because our ancestors had only one flagellum. Plants, chromista, so red algae, green algae, the chromists, which is diatoms, foraminifera, so a lot of the phytoplankton, kelp, they are members of Bicontus. They have two flagellum, or they're descended from an ancestor of two flagellum. There's some debate over whether or not excavata, which is there were basically three groups of eukaryotes. There's excavata, uniconta, and biconta. Now the question is excavata actually a clade? And it includes like euglenozoans and trypanosoma and a couple other weird guys. Trypanosoma is what causes African sleeping sickness. And so there's a question of, well, first, is it a clade? And if it is monophyletic, is it more closely related to the biconts? Because a lot of excavata members have two flagella. And if it is more closely related to Bicontus, so they form a branch together, or is it basal to both? Is it basally derived? Sorry, with respect to both, not basal to both. And so that's there are some questions about it because protists are kind of interesting. They have they evolve very rapidly, right? They have very short lifespans. They evolve very rapidly, and so figuring out who exactly is related to who can be a bit of a problem.
Yep. So yeah, it's always a little bit of a guessing game. Does this belong in this clade or this clade and all of that? It's not always so black and white as some creationists want it to be.
Right. I mean, and the world would be so boring if it were. Creationists are just RJ. I don't know if you're aware. RJ and I have a book titled "The Rocks Were There." We well, if you aren't, RJ and I wrote a book. We published it in February. It's titled "The Rocks Were There," and it's all about how young Earth creationism screws up all across the board. There's actually volume 1 of 2. We're gonna have a second one coming out eventually, and it's a really thick book. But we discuss all sorts of ways creationists screw up when it comes to science.
Yep, and speaking of that book, I got the Kindle version of it right here. I started reading through it, and it's a fascinating book. I strongly recommend that you guys read it. The Rocks Were There, Straight Science Answers to Creationist Questions, Volume 1. Lots of great stuff. There's a Kindle version and a paperback version, I think. And
yes.
Very, very good book. And once I'm finished reading it, we will be. I will hopefully have Jackson and RJ Downing on to discuss the book and have a little bit of a book club.
Yeah, we're very proud of it. We have lots of charts, lots of things that. This is the most comprehensive creationism refuting book, as far as either of us have ever seen. We cover far more claims. Far more subject areas. It's kind of like, if they made Talk Origins into a book.
Yeah. And for those interested in geology, I strongly recommend this book here, The Grand Canyon, A Monument to an Ancient Earth. One of the top claims from creationists is that the Grand Canyon was laid down by the flood somehow, which of course is impossible. But this book is fascinating. It really takes you through the geology of the Grand Canyon. I honestly have never been a geology person. I just find geology to be boring.
It's again, that's one of those topics. Yeah, it's kind of interesting from a distance. I read geology papers rarely, very quite rarely. I read paleontology papers a lot more, and of course, they have aspects of geology. But just a geology paper, I don't usually read those.
Yeah. Hey, Leo Felius is here. Hey, Leo, good to see you.
I think, was that him walking off?
Yeah. Well, he'll be here in a minute. I'm going to remove him until he comes back. Okay, here we go. Hey, Leo.
How's it going?
Hey, Leo.
Hello.
Thanks for joining us. So, we're still waiting for Sal to get back.
Where'd Sal go? Is he doing something? Is he taking care of his mom or something like that?
Yeah, he went to take care of his mother, but hopefully he'll be back soon. But I just wanted to get started that way.
I need to say really quickly, Jackson, Weedy boy. I'm sorry, I had to say that one time. It is an honor to meet you finally in person. I've watched a lot of your videos. I did recently. Somebody I read somewhere, or did I see in a video somewhere with you something about that you had a debate with SFT, and ever since that debate, he's like done everything in his power to avoid you. And I watched that debate. Wow, that was. I mean, it's if you think about it, it's really not hard to defeat the bullshit that SFT says. But
that's fair.
You just there was not a single personal attack like he claims. There was just nothing. It was nothing but this is the data. This is how we know it's true. Now,
does he claim their personal attacks?
I believe he has in the past claimed that like you committed ad hominems and straw mans and all these other things.
I have no animosity towards standing. I have no problem. We've had multiple chats. Well, not since then, but we. God, was that was like a year ago, wasn't it? But
I watched the one on modern day debate, and I think he did very, very well. You and Walker, just a walking fish. I don't know if you know who I'm talking about.
Yeah,
he recently had a debate with him. You, him, and Erica are three of the best debates I've ever seen against him. Just because you guys, all three of you know exactly what you're talking about. He'll bring up like some point that he thinks makes his case, and then you're like, well, no, we know that's wrong, and this is how we know that's wrong, and then there's just going off about something completely different,
Well, I appreciate that, but yes, you're right. He, I have no problem. I have no problem with him. That was back when I kind of viewed him as someone worth talking to. I think, because I went into that debate and we pre-wrote the script, of course. Right, we pre-wrote the script, right. And sort of like, have you ever seen Matt Dillahunty and Seitan Bruggenkate?
Oh, I have not. I need to watch that. I keep forgetting about it, but
it's my favorite debate of all time, simply because Matt wrote his rebuttal in advance because he knew every single claim that Cy would make, and so my scripts. So I write a script and then I send it to Evo Grad and Nestlig and RJ, and they make sure everything is right and all that, and edit it. And so we're working on it, and we decided we know everything he's going to say. Why don't we just rebut it in advance?
Yeah, actually, it's funny because right after the Walker debate, Standing for Truth emailed me and wanted to do a debate with me on Neanderthals, and I already have my rebuttal written out for him.
Do you know why?
Yeah, because Walker beat him so badly.
Yeah, Standing likes to have debates with people he thinks he can use the technical knowledge, or not technical knowledge, technical jargon to confuse them, to make it look like he's winning, so that he can then use him as an example of like how big brained he is when that's not the case. I did want to say really quickly, the best debate between any theist and any atheist that I have seen as of yet is the one between Sean Carroll and William Lane Craig back in, it was either 2013 or 2014.
Oh, yeah. I love that debate.
I remember that one.
Oh my God. Sean Carroll and I don't like, using this terminology to talk about it, but Sean Carroll just whooped him from one side of the stage to the other intellectually the whole time. Just like, no, this is wrong. This is how we know. Exactly. And the one part where Dr. Craig was trying to correct Sean Carroll on the model that Sean Carroll wrote twice. And the third time, Sean Carroll was like, I would know, I wrote the model.
Yeah, it's like
it was
it's like Bernie Sanders when he stood up on the Senate floor and said, I wrote the damn bill.
Yes, it's like they're trying to tell me about what my bill says. I literally wrote the damn bill.
Yeah. Anyway,
well,
so
are we ready to jump into the?
How's it going, Sal?
Sure. So that being said, let's get into.
Hold on. Say something really quick, Sal.
Sal, you're kind of.
Hello.
Yeah.
How's the audio?
You're lagging a bit.
You're lagging a little bit.
It's improving a little bit. The bit rate would just kind of.
All right, I'm going to shut off my camera. Let's try this.
Okay. Yeah, I think that'll help.
Without the video?
It's good. It's good.
Okay.
Yeah, the bit rate is just reduced with the camera on. Yeah.
I can turn mine off if you want me to.
It doesn't matter. I'll probably keep my camera on.
If I could.
So let's
Just
just
say
get
hi
into it.
to the people joining me on my channel.
Hello, there's
I want to say hi to everyone at the Evidence and Reasons channel. David
Hi,
Neff,
guys.
David Neff was very gracious to do a simulcast. So greetings to everyone on the Evidence and Reasons channel. So one reason that I do this, it'll also give people that are watching this an idea what it's like to be a creationist going through school and having to associate with people that don't agree with them, to have classmates that are highly intelligent and educated, who are both biology students, biology professors, and it's a very different environment. And so it's normal for me to feel in an environment where I'm surrounded by non-creationists, and so this is these are the people that I have to engage with when I go to school. And if they ask me my views, which is rare, I have to be able to explain it to them in their language. I have to say it's a different feeling. I've done this before where I've had to speak in front of a crowd of postdoctoral students. These postdoctoral students are students that already have their PhDs and they're going in to study more. It's different being able to make a presentation to that crowd than a crowd that's from the church. So this is an excellent venue for me to practice and discuss my ideas. Course material for, and I'm just showing you some pictures here. I'm trying to develop some course material. People in the creationist community, particularly those who are also science students like me, who are struggling with the same issues I did when I was going through school, and also now professionally trying to go deal with academics who don't share my views or who are even hostile to it. So this is, I know for some people that go to creationist channels and it's like, well, Sal does business differently. It's like, well, I'm giving you a picture of what business is like for someone who's a professional in the field of creationism that actually has to go to universities and go to research institutions and to deal with people that are either hostile or, they're not creationists. It's a totally different ballgame when you have to talk to people that are really skilled in their profession. So this is what it's going to be like, and I'm hoping to equip those Christians and creationists who are going through science curricula to reassure them they're not alone, and maybe they could see it from my perspective. If there's anything I can say that can help them, I hope I can be of help, and that's about it. So, David, thank you for letting me speak to the crowd there at my channel. It's very kind of you.
and thank you so much.
Welcome, Speed, my good buddy.
Welcome,
Hello,
Speed.
hello, hello,
So
hello.
yeah, so this kind of is a little bit of a spillover from last night. Sal, Leo, and Jordan and I had a really good conversation on dating methods.
That was such
Very-
a good conversation. Very respectful. That's why I
Oh, yeah.
like Sal.
Yes, we all like Sal here. So I guess now we'll get into the meat of the discussion, which is
I
eukaryotic
like you guys
evolution.
too.
Well, thank you.
Woo!
So I guess I did prepare a little bit of a slide for abiogenesis real quick.
And I just wanted to comment to that effect. Most of my knowledge, I'm not going to say expertise because I'm not an expert in any damn thing, is probably going to be related to abiogenesis and origins of life. I've done most of my research in that field. I know a bit. I know enough about evolutionary theory to know that it is correct and why it's correct. But in terms of getting into the minutia of it, that I'm going to lead to both David Jackson, and Speed, because they all know infinitely more than I do.
well, thank you. Let me just try to pull this up real quick. So, so a very quick presentation. So I guess what are the characteristics of life? Well, all life has a genetic code that can replicate and pass on to offspring. All life has the ability to respond to the environment, maintain homeostasis, and are basically made of cells. So first, let's understand what abiogenesis is not. Abiogenesis is not us coming from rocks, as Kent Hovind likes to say. So yeah, Kent Hovind has a line that he repeats quite often.
Rock soup.
Yeah, rock soup. But of course, nothing could be farther from the truth. The real theory of abiogenesis is simple chemicals eventually were able to polymerize and all of that. So this is kind of some of the fun stuff that we're going to talk about. First is the famous Miller-Urey experiment, which stimulated conditions on the early Earth. It ran for only 1 week and produced a significant number of amino acids, the building blocks of protein. And what's fascinating about the Miller-Urey experiment is that they didn't try just one cocktail, so to speak, of early conditions, but they've tried several different combinations, all of which produced amino acids. And here we have the a meteorite that fell in Australia in the 1960s, which is actually dated to over 5 billion years old. So this is actually older than the solar system. And it was actually found to have very complex molecules such as amino acids, alcohols, the aliphatic hydrocarbons, and the hydrocarboxylic acids. So lots of really complex things happening in space. And now, a few months ago, I think it was, there was some evidence of the RNA world hypothesis, which is the RNA world hypothesis is the hypothesis that I personally subscribe to. I don't think we're anywhere near a complete understanding of it yet, but Thomas Carell actually did an experiment where he was able to generate RNA bases in the lab. And so yeah,
Can I add just one really quick thing into this? So, because you mentioned the Miller-Urey experiment, which I think was a really good experiment, but I did want to add that one of Stanley Miller's graduate students, I think his name is Jeffrey Bada, I think it's Bada. That's what I assume. I might be wrong. He worked with Jim Cleaves at the Scripps Institution of Oceanography at the University of California, San Diego, and what they performed experiments that were very similar to the Miller-Urey experiments. But what Jeffrey Bada noted was that when they considered what the atmosphere of the early Earth would have been, there was a lot of carbon and there was a lot of nitrogen. And what this does is it creates nitrites, and those nitrites would actually destroy amino acids almost as fast as they would form. However, they performed the experiments that were similar to what Miller and Yuri did again, but they added iron and other carbonate minerals to the mix, to the soup, the mix, whatever you want to call it. And what they found is that when they did that, the products were rich in amino acids, which suggests that yes, we can still get amino acids on a prebiotic Earth that is very high in carbon dioxide and nitrogen in its atmosphere. I did want to add that. I also wanted to add that in, I think it was 1962, Jean Oro, I might be pronouncing that wrong again, was able to produce with ammonia, I think it was, and hydrogen chloride. He was able to produce large amounts of adenine, which is one of the nucleotides. So we have created not just amino acids, we've created nucleobases and we've created nucleotides. So we've done actually quite a bit chemically.
Yeah, and what's really interesting is and this is all in less than 80 years. Mm-hmm. Imagine what we could come up with if we had billions of years to do it. And we know that amino acids form in space. So amino acids form no problem whatsoever. So again, to me, if you know anything about biochemistry, abiogenesis isn't that hard of a pill to swallow.
Well, I have a slightly different take.
Okay.
I would be willing to bet that making organic molecules is easy, but I would also be willing to bet that making life itself is difficult.
Oh yeah, definitely.
That's my 2 cents.
Oh yeah, definitely. Yes.
Because I know that there are some people who are in the who are in like the, oh no, it's all over the place. Yeah, I don't really think so. I think it's probably pretty rare.
Yep. And this is one of the reasons why I love science so much, is that we're faced with a complicated question. Where did life originate? And that's a very difficult question. And it's a great topic for science to explore. And if science cannot explain something, then neither can religion. I think it's dishonest to say to know for certain what we cannot know for certain. And I think the most honest way is saying, I don't know, therefore let's find out. Sal, do you have anything? Sal or Steve?
I was just about to say I do think Sal probably has some objections he'd like to give.
Oh,
I would
course,
be interested in hearing them if he's got any so far.
Yeah, sure. So Sal, I'll let you have plenty of time to respond. Yeah, Sal, you're frozen.
His camera's frozen, but I do think there's a bit of a delay between us and him.
Yeah, Sal, I think it's probably best to turn off your camera.
Yeah.
Here we go.
Now, please unmute my camera. The Yuri Miller experiment is to represent that as in step 4, which was the belief in the 1950s. That's not very accurate. In fact, it's pretty terrible, because we could take a frog, put it in a blender, run the blender. What remains of it will have far better quality of amino acids in the homochiral state, plus carbohydrates, nucleic acids, and lipids. And that's not going to reassemble. No one expects it to become living. And we could do that even with an individual cell. So to be, I'm not taking this out on you, David, specifically, but for the whole industry and textbooks to keep representing this as some sort of significant step when it's known in literature, materials like this will degrade. Not to mention, there is, what I'm showing here is the topoisomerase enzyme. We'd be dead without this. And for the topoisomerase enzyme, and it does, but there are many, let me show you. And for those of you who've had biochemistry like Jackson, there are many enzymes that have quaternary structure like this. This is a homodimer of topoisomerase. What that means is there has, in this situation with a homodimer, there are 2 exact copies of this sequence to make this. So the red and the blue are identical. The odds of something this large, or even say 100 amino acids forming any of these symmetric kind of enzymes, it's astronomically remote. That's a very simple probability calculation. Now, the reason that cells are able to continue is they have a genome. So the Yuri Miller experiment had no way of preserving the recipe of the sequencing in order to recapitulate the proteins. Because if proteins can't be recapitulated, there's not going to be any life if the recipe's gone. So that step in the earlier graph that you showed, David, where it was a series of these steps, this is the fact that there's no genome in many of those scenarios. It's an automatic dead end. And this is finally acknowledged, thankfully, by origin of life researchers themselves, and even a top evolutionary biologist by the name of Eugene Koonin, who's not, who really doesn't delve into origin of life, but it bothered him enough. And he was commenting on the RNA world and its problems in creating a translation system that involves either DNA and or RNA serving as a template for proteins. He said the odds are so remote, we need multi-universes. So these are not creationist probability arguments. These are probability arguments and concerns that are raised by the community themselves. So I appreciate the enthusiasm. And yes, I do agree that we may not know. So there's one side that believes that science will figure this out. That's a statement of faith. And it's also a statement of faith to say that it originated naturally when everything we're observing and the more that we study indicates that the problem is getting worse. That's why we went from the situation with Yuri Miller where people like, I believe it was Simpson saying we're so close to solving the origin of life problem, now to the point where we have people like Eugene Koonin having to appeal to multiverses, which we cannot see, which we cannot test, which we cannot prove. And it's just, at this point,
I'm
really
almost willing to bet money
so
that you are misrepresenting what he's saying. As someone who has read a number of papers by Eugene Koonin, I'm almost willing to bet money that what you're claiming he's saying is not what he's saying. I would actually, if Sal, would you mind putting the link to the paper that you're referring to in there? Because I don't disagree with you when you say there.
Yes, and I'm a betting man. Well, actually, it's illegal to.
That's
to
what I
wager
said
on the
almost.
internet, so we don't wager real money.
I'm actually in agreement with you.
We'll wager Monopoly. I'll
$1
wager
million
you
Monopoly
$1 million
dollars. Okay.
in Monopoly.
Well, look,
okay.
Oh God,
I
Speed's getting up to get his books.
Okay.
Are there problems with the RNA world model as it stands currently? Yes, as it stands currently, yeah, there are.
Can you Google eternal inflation in Koonin? If you Google eternal inflation in Koonin, just Google eternal inflation in Koonin.
Might I ask what the eternal?
Go to the appendix. He'll provide a calculation for RNA replicase.
Hey, Sal, it's kind of hard to hear. Am I the only one having trouble hearing Sal?
I can hear him just fine.
Okay. Sneed, I think you have a little thing to show, if you're ready for me to show it.
Not quite yet. Let's see.
Might I
on the Koonin thing, basically, Eugene Koonin wrote a whole book about the nature and origin of biological evolution. And in this book, there's a couple places where he waxes eloquent about how difficult the problem is. You know, in his RNA world hypothesis is something that Demeter really pushes back on. And Koonin pushes back on it himself. But one interesting thing is in this same book, he calls intelligent design malicious nonsense. That's in his appendix. So, I mean, there are other quotes in this book that you should probably read along with the one that you picked out. And I found the cosmological inflation part. But anyway, first of all, I wanted to push back on what Jackson said. I think, actually, I used to agree with you, but now that I've started to look at a little bit on prebiotic Earth, on inorganic chemistry, on clathrates and minerals and small peptides and all of that, and exobiology, I think actually life is ubiquitous in the universe. I think anywhere you find an Earth-like temperature range, I think you're going to find something is going to have happened.
You're right.
Speed, I agree with that. I can agree with that.
Well, I don't know if I'm right, Jackson. I don't know if I'm right, but I swing from one end of that pole to the other. I mean, I'll start to find something. But anyway, I do want to. Now, I want to point out that what you're talking about, Sal, is protein probabilities, the probability of creating a topoisomerase, from scratch. And Douglas Axe is a creationist, I believe. I don't know if he's young.
He's an intelligent design proponent.
Intelligent design proponent. And he wrote this famous paper that came up with, there was 1 in 10 to the 72nd, I think, or 74th proteins that would be functional. He came up with that estimate, which was done with not a lot of information. But the problem is that the space that he's working into was 20 to the 150th, which leaves an amazing amount of functional proteins. And people quote that paper, but go ahead and share my screen.
Okay, sure.
Really,
Douglas Axe-
really quickly, after Speed is done, I do want to give Sal a chance to respond to both
Oh, yeah.
Jackson and Speed, because I do want to give him ample time to provide his objections.
Oh,
Okay.
sure. Go right ahead. And
Oh, I mean,
Sal,
I have other objections to what he was saying. I mean, he mentioned throwing a frog in a blender. That's just a
yeah. By the
straw
way,
man. No one really cares if you throw a frog.
by the way, Sal, if you feel that you're being if you're being overtalked, or if you feel that you want to say something, please don't hesitate to chime in and state something in the private chat. I want to make sure that you feel comfortable, and I want to
Okay.
make sure that you're.
Oh, no, this is okay. I mean, this is something that we should have
Yeah.
probably talked about before, and it's no fault of anyone. If anything, it's a fault of
Yeah,
mine for not being assertive
Did
enough.
I overtalk you, Sal?
I would have requested a little more equal time because I do have a channel. I need to, because this is simulcast to a channel, my listeners would be
Oh, of course.
wanting to hear my side of it. So if it weren't simulcast, I think this is, so first apologies. I'm not blaming anyone. If anything, it's my fault. I should have tried to square that with you first, David. So, but there are people on my channel that want to hear about equal time. So I don't know how we're going to
Okay.
manage this. And I feel kind of bad because we have Leo here, Speed and Jackson, and it's like, I don't want to, I don't want to silence them either. It's not
Oh, of
exactly
course.
fair that they be silenced.
Okay,
They've taken the time to be here.
Did I overtalk you, Sal?
So all I could ask is, can we have a little more equal time? And that means you guys will not have as much time to talk. But because we hadn't figured this out at the start, if it's going to be equal time for everyone, that's fine. We'll just know better next time. So I'm
Yeah,
not
and I do
faulting anyone for
apologize
how it's
for
going.
the unbalance. Sal, feel free to invite any of your creationist friends in here, so you're not the lone creationist.
I would much appreciate if Standing for Truth.
Actually, I'll clone myself. I'll just clone myself and
Okay,
I'll just
sure,
speak on
and
their
that's not a
behalf.
problem. And
Okay.
I know I probably should have done a little bit of better job hammering out how much time everyone was going to get beforehand. So again, Sal, don't feel free to say, "Hey, I want to speak up a little bit more." Don't be. I want to make sure that everyone feels comfortable and respected. So Jerry says, "Is this a debate?" No, this is more of an informal discussion on these topics. But Speed, go right ahead. Floor is yours.
Really quickly, I just wanted to say, if somebody like Speed makes a point and then Sal wants to say something to that, he should have the time to say what he wants to say, and then maybe Speed give like a really brief back and forth with anybody that makes a point Sal wants to object to. That way we can ensure that he's getting equal time, but also anybody that wants to object to him also has a moment to do that. I think that would be fair. Go ahead, Speed.
Okay, I want to point out that y'all overtalked me, and Leo, you started it.
I'm sorry.
So bite me. Anyway, okay, I was making a point, and I only have about 1 point to make, so I'm not going to need a lot of time, so please give me this. But Douglas Axe wrote an earlier paper where they also did some stuff with proteins, with the protein space and what is functional. And he said, okay, they took this Barnase and they modified it. They mutated all the cores in every which way, and they found out they did all sorts of hydrophobic alternatives. So they cut out different domains using a sensitive biological screen. We find that a strikingly high proportion of these mutants, 23%, retain enzymatic activity in vivo. So this is a paper. In the other paper, it was 1 in 10 to the 72. Here he's saying 23% were functional. So I mean, you got to look a little deeper into this functionality and the protein space. And that is my point, Sal. Nobody has suggested that these proteins occurred one amino acid at a time and formed a fully functional 1,000 amino acid protein. And I am done.
Okay, Sal, I'm going to give you as much time as you need to respond.
And by the way, gentlemen, I really appreciate the civil discussion. This was
absolutely.
Again, I take some responsibility for how this is going, and I really do appreciate you showing up. And just just so people understand, when I work and go to school, I have people like Speed of Sounds for my colleagues. Someone like Speed of Sound or Leo could be my lab partner in chemistry class. So impersonal against them here. And they're just great. Leo and I hopefully will study quantum mechanics together sometime in differential equations.
For sure.
And maybe Yang here will be studying molecular phylogeny. So specifically with, first off, Jackson's paper, I linked it in my
I'm reading
other
it.
channel, and I'll tell you why. I'm so slow on the computer that I'm on.
I found it.
And that's
I found the paper, and I was looking at that.
You can go down to the bottom where it talks about the RNAs, and you'll see the probabilities
Yeah,
there
I've been
at the
looking
very
at it.
bottom on eternal inflation. Regarding Speed of Sound's discussion on the AX paper, those are monomeric proteins. The enzyme can have enzymatic activity with just a single. It doesn't have really a quaternary structure where we need multiple polypeptides to form it. It's a totally different ballgame when there needs to be these dimeric, tetrameric, or whatever, or hexameric proteins to be able to enable enzymatic action, such as either a tetrameric helicase or a homohexameric helicase. And the reason that we have these proteins that have these multimeric structures, just like we have it in engineered systems where we have nuts and bolts that are symmetric, it's a very nice way to create symmetric systems. So even though I would agree we could preserve some activity, it doesn't mean it's going to evolve into something complex. Just because a tornado passing through a junkyard can make paperweights doesn't mean it's going to make a 747. So there's no question, and this is something that I will take issue with some creationists. You could take a polypeptide, it's going to be able to have some chemical activity. So it could be like equivalent to a paperweight, but just because we could find some chemical activity or catalytic ability in some random polypeptide doesn't mean it's going to be able to be evolved into a complex protein. And also, even if we have something that's functional, doesn't mean it's going to be the ancestor to functional proteins. That was the problem that I had posed about a month ago, and I said, do all proteins evolve from a single gene? And that was the actual point I was trying to make. Just because you have something that's functional now doesn't mean it's going to be able to evolve into something else that is also
do have
functional.
one really, really quick, really brief question for Jackson. Do they state in that paper the method by which they determined the probabilities that Sal is speaking of?
They go in the appendix. It says probabilities of the emergence by chance of different versions of the breakthrough system in an O region, a toy calculation on the upper bounds. But down here at the bottom, it says the model considered here is not supposed to be realistic by any account. It only serves to illustrate the difference in the demands on chance for the origin of different versions of the breakthrough system, and hence the connection between these versions and different cosmological models of the universe.
Okay. Which makes sense from a statistical analytical standpoint.
Yeah.
Jackson, I wanted to give.
The point is that it simplifies the problem. The problem is far worse. But the point of the paper is he's arguing for the MWO, many worlds.
Right.
Some sort of many worlds
eternal
hypothesis.
inflation as you continue to increase the amount of planets and worlds necessary or that exist, then it becomes essentially inevitable that life will result.
Yeah, I tend to be.
Well, then that means we can live in a world. I mean, that means there could be a world where the creationists are right. This many worlds solution is. It's like, well, there could be worlds where the creationists are right, and there could be worlds where it's not. And what world are we in? So if anything is possible,
that's what they're necessarily saying either.
It's not necessarily. Not only are there philosophical problems, there's an issue of whether this is science, because if it's not testable,
I agree, but
really
to be fair, you brought up the paper. You brought it up, so I don't understand why you're going to argue with the paper you brought up.
Yeah, but I tend to agree with Sal on the many worlds hypothesis.
Oh, I'm
Um,
not saying I agree with it either.
Yeah. So anyway,
The physicist in me is coming out because the many worlds hypothesis is different from a literal multiple universes model. The many worlds hypothesis simply states that there's one universe that occupies different timelines. We can represent branches in that timeline by wave function collapse. It's the same universe, it's just that the timeline of that universe can evolve in different ways based on how the universal wave function evolves. So the Everettian model of quantum mechanics, or the many worlds interpretation of particularly quantum mechanics, does not postulate other actual separate universes. I just wanted to make that distinction quickly.
thank you, Leo. Because I think it'll be fun to do a stream just you and me, Leo, on the multiverse and stuff like that. Because that's stuff that goes
For
way
sure.
over my head.
Could I get a word in on
Yeah,
that?
sure, go right ahead, Steve.
Kunin. Kunin is basically a biologist, and he's fascinated with this many world hypothesis. And at the end of his book, he kind of went off on a tangent, as people do when they're out of their field, out of their depth. And that was not. It has very little meaning what Kunin thinks about RNA world or whatever. He's
a
one
very
biologist,
intelligent
basically,
biologist, but
and he
just
did a lot of work.
Yeah.
He is, but he's a little whack when it comes to the many world thing, and he wrote about it in his book too. So you can't use that to represent the field of abiogenesis. One person, his RNA world didn't work out, and he's like, okay, well, maybe many worlds will save my ass. But no, that is not a valid point. What Eugene thinks.
Okay, sure, Sal.
Okay.
Back to you if you want to say anything.
I was not endorsing Kunin's work or his many worlds hypothesis, except to say the probability is very remote. And this was not the sentiment in the 1950s when the Yuri Miller experiment came out. That's why sometime thereafter there were all these extraterrestrial theories and then all these other theories that kind of came out because people began to see the problem of not having a genome. The Yuri Miller and these prebiotic systems were just going to be insufficient. And not only that, the polymers will degrade. If I could point out where I've gotten a lot of my material was
by all means
curated by a creationist who actually works at a secular university. She was a postdoc at Harvard. Her co-author, Rob Stadler, the name of the professor's changed hand. She's at University of Missouri. She teaches molecular biology. And her co-author is a PhD at Harvard, MIT. And so these are, I read the book and my jaw dropped. And when she teaches her molecular biology course, she will cover these issues, not in a very overt way, but she's just pointing out, okay, this is what the professional literature says. And so nothing of what I said about saying it's highly improbable or that there are no solutions now, when I'm representing it as becoming worse over time, the actual literature, segments of it, maybe the majority is optimistic, but there's a minority that's skilled, like say Steve Benner, that think this is just like going nowhere. Unless we find new laws of
the protein biologists like Liam Longo and Martin and Nick Lane and these various other biochemists who are working on it and are convinced that it is going somewhere, you just tell them, no, you're wrong.
Yes,
so
I'll tell them they're wrong.
So despite the work that they have done,
James Tour called them out. James Tour called them out on it.
Yeah,
and not only that,
from
Steve Benner is their
the walls
peer,
of the Discovery Institute. You know, but regardless, but I do agree with you, Sal. I'm not really. I don't agree with this paper either. So we're in the same boat. We both agree. How about that?
How about that? So actually, when we discussed this in the email, Dave, and we're informal.
Yeah.
I was
Sure,
going
yeah.
to
So
talk
let's go
about
there.
eukaryotic
I
evolution.
was just about to kind of shift the topic to eukaryotic evolution because we've been kind of like in a million different directions already. So I guess eukaryotic evolution. I'll let Sal. I'll let you have the floor about eukaryotic evolution.
So there are various ways that creationists have attempted to critique universal common ancestry. And I'm sorry, I'm going to offend some of my creationist friends, but I think going after chimp-human common ancestry is the worst way to be going after this, because we are very, very similar to chimpanzees. I see it when I do gene browsing. I'm not a laboratory person. The gene sequencing guys are laboratory people. Instead, I go after the two aspects of universal common ancestry. One, is there a universal common ancestor for all genes? And Speed of Sound is kind of, I was amazed, he's actually with me on that there's independent origin of all the major protein families. And if I'm misstating Speed's viewpoint, he's here.
Yeah, but you're talking at like a huge level. You've included like all animals, all fungi, all plants at that point. And so if you want to say, if you kind of push back to the point where you're arguing about, okay, well, the translation enzyme machinery between archaea and bacteria are different. Welcome to our side. Young Earth creationism is dead. There we go.
So do you believe all genes, Jackson? I mean, what are your views? Do you think all genes descended from genes/proteins descended from a common ancestor, or do they have independent origins?
All of life is ultimately descended from a common ancestor.
In that respect.
Are there any proteins around today?
But how about
are there any proteins around today that are descended from, or that were in our last universal common ancestor? I don't know. I haven't done the research on that. So
Yeah,
I don't know. I suppose I wouldn't be surprised if there weren't any, but I don't know.
Yeah, and I should point out that even if there are independent origins for certain groups of genes and stuff like that, that doesn't necessarily prove young Earth creationism. It doesn't even prove creation. It just shows that, okay, we have a couple of different independent genes that kind of diverged and diversified.
Right, exactly.
So we can't make a leap from saying, okay, there's a couple independent origins here, therefore God.
So it's like Sal, if you want to say like, well, we don't have cytochrome C in common with bacteria. Okay, I don't know if that's accurate. But that's just an example. Okay, fine. But we have cytochrome C in common with hippos and mushrooms and oak trees. And so that's not really an argument for either intelligent design or young Earth creationism. They're both way, 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 way out of the way at that point.
So, Sal, have you ever heard of the phylogeny challenge?
Well, I think Speed was trying to say something.
Yeah, sorry. Speed, go for it. Speed, if you want to say something, go right ahead.
Okay, that conversation first occurred on my channel with Sal, where he thought it was like amazing that I don't believe that all proteins had a common ancestor. I mean, what happened to us?
Okay, hold on a sec. I think
okay.
Yeah.
I thought we died. Anyway, of course, all proteins didn't have a common ancestor. Are you still with us?
Yeah.
Yeah, we're good. We're good.
Yeah.
I have no idea why he keeps beach balling on me. Anyway, so basically the idea that all proteins had a common ancestor to me is absolutely insane. I mean, there was an ocean full of basically small peptides forming before life happened, and there were probably a number, like trillions of different cells that formed and popped up. And in these, there were many, many different protein ancestors. And then not only that, but when you when you start going forward, proteins don't exactly reproduce like organisms. Sometimes they get chopped up, and sometimes there's different splicing of exons. Sometimes two proteins are smashed together, and you have an entirely different animal. I mean, there are so many different ways that proteins can get shuffled that no one in abiogenesis or biologists should ever think that it had a common that all proteins had a common ancestor. Eukaryotic cells, even, it would be well, when you think about it, it seems like there should have been a common ancestor, but maybe not. Maybe it happened multiple times. So.
Yeah, and that being said, abiogenesis probably also happened multiple times independently. So
yeah,
I don't think there was like one.
That was actually an interesting point by Steve.
I'm sorry.
Go ahead, Sal.
I interrupted you, my computer, so I really apologize.
No, it's not a problem.
Dave.
Go right ahead.
You're good. You're good. Go ahead.
Mm-hmm.
I wanted to say that was an interesting point by Speed. He and I'd like to understand what his views are. Does he think possible the eukaryote had a separate origin than prokaryotes? That is somewhat similar to things. I actually, it's hard to pin down what Carl Woese actually thinks. I just wanted to hear what Speed of Sound, because he said that he thinks that lots of cells popped up, millions of them, and I wanted him, if he could clarify, Speed, I'd be interested.
Basically, in these in these huge pools of organic chemicals, the formation of a cell was probably something that occurred millions of times in different ways. That's not to say that there isn't one common one that took over at some point. We don't know that, but I mean, there's also a possibility that a number of different pre-prokaryotic life forms developed all at once. You have a massive pool of biological chemicals. So if you understood what I thought, you would no longer be a young Earth creationist.
Okay, Sal, are you trying to share your screen?
I don't know.
I will. We're just having some slowdown
I wonder if it's
problems.
because of your simulcast.
Maybe I'll stop sharing it.
I wonder if your simulcast is causing some issues.
Well, he's not botting too much, so I don't know.
Yeah.
He is botting a little bit. I'm multicasting.
I was just trying to let everything catch up.
Okay.
So in Speed's view, there's a possibility that there's independent origins of major cellular families or architectures, like, say, eukaryotes and prokaryotes.
I wouldn't.
By the way, that seems to be. I mean, other people.
Well, there are groups, or there are certain proteins like chaperonins that we share with archaeans who are prokaryotes. So we have genes in common with archaea. That's how we know that the Loki archaeota are the closest relatives of eukaryotes. But sure, what he's saying is some of the genes, or some of the proteins, are independently derived.
Yeah, we have to separate. We're talking about proteins having a common ancestor, and we're talking about cells having a common ancestor, and then other organisms. But the mechanism that caused the first eukaryotes, the invasion of what was it, archaea into prokaryote, that could have happened a number of times. I mean, that could have been a certain type of cell that had replicated where that was very common, and there could have been a number of eukaryotic cells that were the origin, and we just don't know. I mean, the complexity back then, even now, the complexity, we don't know what is going on out there with the prokaryotes and the archaeans. There is an immense world yet to discover, it is premature to pronounce it all dead.
I just shared
absolutely, yeah.
I just shared 2 links in the private chat. The first one is more simplistic. It's from evolution.berkeley.edu. The second one from genomebiology.biomedcentral.com. Actually, that paper talks about how eukaryotes may have evolved out of prokaryotes, and it lists numerous different ways that this could have potentially happened, but it really focuses on endosymbiosis as being probably one of the most likely ways in which this occurred, since that's sort of on the subject that we're talking about, and that does relate to what Speed just said as well. I did see
Could
that.
have
Right.
happened a billion times.
To Speed's point, I mean, we only knew the first paper to describe archaea was written in 1984 by Lake et al. Right. That's how it, within my parents' lifetimes is they within their lifetimes is how long we've known about an entire domain of life. We know very little about this group, and then the first phylogenies to come out, you had the eocyte hypothesis and the 3 domains hypothesis, and these were sort of combated each other for a while, and now we have even more and more groups. It turns out archaea is highly paraphyletic to eukaryotes. There's so much we don't understand about archaeans, and it'll be interesting as more data comes in future years.
And the thing is, as symbiosis, basically we have 2 independent bacteria. One bacterium engulfs the other, and there is a beneficial agreement. And so this gets past 1. And the interesting thing is that what is mitochondria? The powerhouse of the cell. Everyone remembers that from
The powerhouse of some cells.
Yes,
The some cells, yeah.
Yeah, it's the cells that have it. But the interesting thing about mitochondria is that it has a small amount of their own DNA.
Exactly.
So the question is, how does the creationist model explain why mitochondria has its own DNA?
It's either mcDNA or it's mtDNA. I think it's
mtDNA.
Okay, yep, that's what I thought. I always get those confused.
Yeah, so it seems that the origins of the mitochondria must be as a result of some form of endosymbiotic relationship where a bacteria swallows another, and there's a beneficial arrangement. So again, Sal, how would you explain why the mitochondria has its own DNA? Yes, I'm here.
David, are you there? Okay.
Yep.
If the Earth is young,
He's roboting.
I mean that, and the point is creationists don't have to explain. It may well be that something under explanation was the cause of it. What I'm criticizing is to assume that this can happen naturally and it's within the odds, and that's within science. We can make estimates of the probability or likelihood of something assembling itself a certain way. That's why we can make statements like a 747 passing through a junkyard. It's not going to make a 747. I mean, a tornado passing.
That is
It's not going to make a 747.
analogy.
I'm going to point out one thing here. This is a graph.
Somebody's got somebody knocking at the door.
This is a graphic of.
Sal, are you there?
Or yeah,
He's there. There's just a bit of a delay. So after I speak, we'll give him some time.
okay. I'm gonna try to cut
Yeah,
one channel.
you might have to cut your
Right.
simulcast.
Let me cut one channel here. All right, I'm shutting down one
Okay.
of my computers.
Google Scholar.
I did want
See if
to
that
take
improves
this
the
time.
speed. I'll shut down another computer if it does.
I didn't want to take this time. I would love to see a conversation between logical, plausible, probable, and Dr. Dan.
Go ahead.
Oh, same here.
LPP seems to think he knows more than an actual PhD in this when he has literally no degrees relevant to anything that we're talking about.
Yeah.
So am I caught up? Is my
Yeah, you're
sound
good.
Yeah,
catching
you're
up
good.
now? All right. It may be when I try to share my screen that things blow up, and I've had problems on this computer. So what I may do next time is use my other computer. So this is a hard lesson learned, and
Where did
God willing, we'll
Where did
figure it
Jackson
out.
go?
Jackson, it's great
I
talking
think
to
he
you.
went somewhere real quick.
So I was showing, so I'm just going to have to just talk through this. The phospholipids in bacteria and eukarya have the same chirality.
Oh, let me-
The phospholipids in the membrane layers of archaea are the opposite chirality. That's one of the problems right there with common ancestry. But there are more problems. A lot of focus on eukaryotic evolution talks about the mitochondria, and I'm just going to bypass that because it ends up being a distraction to some very serious problems with membrane-bound organelles, such as the nucleus. I mean, membrane-bound organelles aside from the mitochondria, such as the nucleus. How is it that we have it that has the genome inside of it and the ribosomes external to the nucleus? So the mRNAs have to go from the nucleus, inside the nucleus, to the ribosomes that are outside. If the nuclear pore complexes are not there to allow this transport, the thing is dead. So that's one of the, even let's just hypothetically assume universal common ancestry, all those models that, and I'd like to thank Leo for posting that, that doesn't solve that problem. So what I see in the evolutionary literature is just focusing on things that they think they can solve and then kind of pretending the other problems don't exist. They don't even acknowledge that it's a significant problem.
Who does that?
So not only is it the nuclear pore complex.
I just gotta say
So-
really quick, shout out to Gutsick Gibbon. She's in the side chat. Okay, I'm done.
Yep. And by the way, since she's here, she's only at like 15 subscribers away from it being at 5,000 subscribers.
I'd subscribe more, but I can't.
So
Yeah.
if there is for some reason you are subscribed to me but you are not subscribed to Gutsick Gibbon, go to Gutsick Gibbon. Excuse me. Go to Gutsick Gibbon's channel and subscribe right now.
I'm going to subscribe to your channel, Gutsick, because you always say the nicest things when I'm on Modern Day Debate.
You haven't subscribed
You're like
yet?
my biggest
I
fan
also,
club
I also need to mention, will Otangelo ever cite anything other than his own personal blog on anything?
I don't know.
I doubt it. Like, I have never clicked on a single one of those reasoninScience.catsboard.com links because it's not peer-reviewed technical literature. It's literally just not relevant at all. It just straightforwardly isn't.
What was this, Leo? I'm sorry.
No, I was talking about somebody in the chat.
Okay, so you said that.
Oh, okay.
Okay.
So I was talking about the problem of the nuclear pore complex.
Well, hold on. What the nuclear what complex? I didn't quite catch that. If you wouldn't mind repeating.
The
Nuclear
pore complex of eukaryotes. How does
The nuke.
nuclear pore complex? This is
Right.
not atomic
Yeah,
nucleus.
yeah,
this
yeah.
is cellular nucleus. And how does something that has a prokaryotic architecture or prokaryotic-like architecture transition from not having a nuclear membrane-bound nucleus without dying in the process? That's a legitimate question. And if the odds are really remote, then common descent needs miracles that are almost indistinguishable from special creation. And that's my point. Is I'm not going to argue against common ancestry. I'm just going to point out, if we assume it for the sake of argument, it's going to take the equivalent of miracles of special creation. Because not only do we need a nuclear pore complex, which also some of these have like 300 proteins that have to be stitched. All of those need signal recognition particles and localization signals, or whatever they're called, to be stitched in the right place, put together, assembled. So you have this complex there. That complex then will recognize both RNAs and proteins that have to have a certain sequence to locate it in the right place. The thing is,
Sal, may I ask you a question? Do you agree that fungi and humans share a common ancestor? If you don't, it's not going to make any sense for your argument.
I'll accept it for the sake of our.
So here's why it doesn't make any sense if you don't, because we share
I'm
nuclear. We share nuclear pore complexes with yeast. We have the same genes encoding the same proteins for nucleoporins that they do, that yeast do. Right? We have homologs of the same genes. So if you don't accept it, this doesn't make any sense for your argument.
I'm afraid that's a non sequitur, because they could be just as easily.
So then God created homologs of the same gene for vastly different organisms. My question to you is, why is God purposefully trying to deceive us?
He's giving you a stairway to understanding. It's really great. We can study fungi to understand nuclear pore complexes.
God wants me to be an evolutionist
versus having to dissect embryos, tear them apart. It's a beautiful stairway to understanding.
God wants me to be an evolutionist then.
Wouldn't you rather be able to study these than embryos and cutting them open? I mean, when I realized that's what they were doing at the NIH, it began to be
so
really
what I'm understanding is
that they had the organs.
So what I'm understanding is God wants me to be an evolutionist, because if God really didn't want me to be an evolutionist, then God would create vastly different genes or vastly different proteins or both. Wouldn't even have to use genes or proteins for these different systems. If we are unrelated to fungi, there is no reason we should have these similarities. This doesn't make any sense under creationism. It does, however, make sense under common ancestry. No, it doesn't.
Yes, it
does. You want to know why it doesn't?
I just pointed out
so
why.
If I ask you,
I just pointed out why.
You just said God could do
understand. It's great. We have these things called. Do you know what a model organism is? Do you know what a model organism is?
Yes.
Do you know what a model organism is? Did they teach you that in school?
Yes.
Okay.
I have learned.
They're called model organisms. We can study human. Yes. We can study human biology by studying other creatures. It's really nice we can dissect fetal pigs rather than human beings.
Why?
Isn't it?
Why do we share?
Huh? Don't you think it's kind of nice that God doesn't have to dissect
why did God
so that people can figure out
why did God give pigs and sharks and yeast the same or a number of the same genes as us? Why? If your argument is your argument really just to help us understand how we work?
I just answered your question because he's sparing you from being dissected. He's sparing humans from being dissected so we could study biology. We can study sharks. We can study plants.
So God decided.
Aren't you really glad we don't have to cut apart
So your argument is God created all organisms in a nested hierarchy.
Asking for an answer.
So your argument, Sal, is God created a nested hierarchy of all life so that we could understand how humans work. That is your argument.
Yes.
Then God wanted us to believe in evolution. Thank you.
So here's the question
because
I have.
A nested hierarchy makes no sense under.
Because
sure. So here's a question I have for you with the nested hierarchy thing that makes it just look like just like what we would expect under evolution. Couple questions. Number one, your explanation is not falsifiable. That God did it just so we could study ourselves. And the second problem with it is that on balance, the nested hierarchy makes much more sense in light of evolution. Nothing in biology makes sense except in the light of evolution. And that is true no matter where we look. And I think the biggest piece of evidence for evolution is just how broad the application is in everything from medicine to ecology, to food science and all of that.
I can speak on the food science. I've literally been to DuPont Pioneer. Well, we used to be DuPont Pioneer. It's now Corteva Agriscience in Johnston, Iowa. I've been to Stein Seed Company in Adel, Iowa. I've been to Latham Seeds in Alexander, Iowa, because I live in the state. And if there was no evolution and what creationists were saying was true, literally everything they've ever done with corn and soybeans could not have happened, but it has. I've watched them do it.
No, I just wanted to,
So someone
what
mentioned
I'm pointing out
the-
is there's common design, and I was pointing out the problem with eukaryotes. You can't, I just gave why a prokaryote would require a miracle to become a eukaryote. Doesn't matter whether it's fungi, whether it's a plant, whether it's an animal, the problem stands. It is evidence against common ancestry being natural. It would require miracles. What ended up happening is you avoided the direct problem of the membrane-bound organelles, such as a nucleus, the nuclear pore complex with 300 genes in them, and then all the reformatting that has to be done with RNAs and the proteins, adding localization signals. So if we share the same genes to whatever extent we share it with prokaryotes, those have to have formatted new localization signals across the board, both for the mRNAs and the proteins simultaneously, plus the nuclear pore. There's no getting around that. So if we're saying it looks evolved just because of phylogenetic reconstructions that don't account for mechanistic problems, like I just outlined, that's ignoring the evidence right there. That's evidence I just gave you. And if you're going to appeal to these phylogenetic reconstructions that are not mechanistic explanations, that
So you
don't deal
heard
with
me
the
when I
mechanical
said-
problems that I just outlined, then you're saying that God made it look evolved. I just gave you an example why it doesn't look evolved, but rather we might consider that the nested hierarchy is there for other reasons. That's one example. That's right at the bottom of the phylogenetic tree. Right at the bottom of the phylogenetic tree is the proteins. They don't share a common ancestor. Someone, I have someone here who just agreed with that. I showed the problem right at the very base of the phylogenetic tree with eukaryotes and prokaryotes. That would take miracles to make that happen. I pointed out why. So to keep arguing just because fungi look like humans, therefore evolution is true, I just gave you a data point.
So
and you
I
totally
wasn't
ignored it.
arguing about prokaryotes. I said yeast, which are eukaryotes. Yes, they are fungi.
Sal, if I can ask a question.
Hierarchy to both fungi.
You're roboting a bit, Jackson.
I'm sorry. Do you want? Am I still roboting?
Yeah, a little bit.
A little less, but still, yes.
Am I still roboting? Can you hear me?
Yeah, you're good now.
Now you're good, yes.
Jackson, are you trying to talk? Jackson, isn't anyone trying to speak?
No.
I'm not.
Okay, so here's a question I have for Sal. Which claims? I guess we can start.
Okay,
with
how do I sound now?
You sound good. Go ahead.
Good.
I sound good? Okay.
Yep, you're good, bud.
So the thing about the nested hierarchy that I'm trying to point out is a nested hierarchy does not make any sense under creationism, under the idea that there are created kinds of organisms. It doesn't make any sense. And the reason is a nested hierarchy is predicted specifically by evolution because evolution is descent with modification. Creationism is not. So it does. The only way it could theoretically only up to the extent that organisms are united by a common ancestry, right? So if a group of organisms would show this nested hierarchy pattern, but would not show it with any other organisms they are not related to, right? Within the Barrowmans. But that's not what we see. We have the nested hierarchy all across the eukaryotic board. Now, once you get into prokaryotes, yes, you get the crossing, you get horizontal gene transfer, and so the idea sort of breaks down. But what I'm talking about is yeast. We share these genes with yeast, the nucleoporins. This doesn't make any sense under creationism. There's no reason for this to be that way. This and as David said, the idea that these other physiological processes and genes and proteins are distributed across organisms so that we can learn about ourselves, that's nonsensical. Because what about the proteins they have which are not in us? That doesn't tell us anything about our own processes. It's totally unfalsifiable.
Which, if it's unfalsifiable, that by definition means it's not science. But I did have a question for Sal. I do believe David brought this up. Before I ask the question, I just want to state that this would be the single largest prediction that specifically the young Earth creationist model would have. It would be the easiest question for them to answer. And that question is, out of all of the cats, if you consider cats to be a kind, out of all of the cats that exist on planet Earth, which one was the one that was originally created by God that all the other cats speciated out of? We could ask the same for birds. We could ask the same for lizards, for fish. That's all I want to know.
I brought a prop. I brought a cat prop.
A pretty prop.
Is that Loki?
That's Loki.
Loki.
Evil Loki.
I don't have an answer.
Hold on. What was that, Sal? Can you repeat?
The structure of nested hierarchies is not an explanation for nuclear pore complexes. The structure of nested hierarchies is not a mechanistic answer for why nuclear pore complexes
No, I
exist.
agree, and I wasn't saying
I'm
it
going to
was.
just repeat it. Nested hierarchies are not a mechanistic explanation why nuclear pore complexes are highly probable. Nested hierarchies are not an explanation for why nuclear pore complexes evolved when they didn't exist. That's the problem with evolutionary theory. It's built on non sequiturs.
I think you misunderstood what I was saying. I was not saying this is an explanation for them. No. I don't know what the origin of nuclear pore complexes is. I don't claim to have knowledge on that. What I'm saying is we share nuclear pore complexes with yeasts, which would make sense under evolution, not under creationism. That's what I'm saying.
It doesn't make sense if it's not going to evolve from a prokaryote. It doesn't make sense under. That's the point. Is if a eukaryote doesn't even evolve in the first place, if a eukaryote doesn't evolve in the first place, it's kind of pointless to be invoking evolution, of fungi and humans when there's no eukaryote to begin with.
I'm not following.
I mean, if there's no eukaryote to begin with,
no,
falsified.
So
as far as the evolution of eukaryotes. So universal.
No, all you're saying is you don't understand it. Therefore, we should accept that you don't understand it. Which we accept you don't understand it. That doesn't mean there is no explanation for it, or that you are the arbiter of knowledge on this topic, right?
So do you think there's an explanation under evolutionary theory why nuclear pore complexes exist?
Is there not? Well, I mean,
and the membrane-bound nucleus. Is there
guys? Call Me Emo has joined. Welcome. Call Me, do you want to say a couple things? I want to give you an opportunity to introduce yourself and talk. Emo, you're muted if you're trying to speak.
Oh, my bad. I may have an echo list.
Did somebody mute him, or did he mute himself again?
Emo, you muted yourself.
Yeah, you muted yourself.
There was a little background noise, but we could hear you.
You muted yourself.
There he is.
Oh yeah, I do that normally. If
a little
if we
background noise, but we could hear you.
Oh yeah, if we speak together, then I live it's kind of like area, so it's always raining. So you're hearing the rain now.
He's got a bit of a delay.
Yeah.
I can't see the screen name now, but you were. Sorry about that. Am I still connected? Okay, sorry. Yeah, I kind of shift location. Sorry about that. So you were asking a question about kinds or archetypes or whatever, and you were asking for the original cat, if Sal could identify that. And to me, that would be a very disingenuous question, because if I was to ask you that question, you won't be able to answer it. If I ask you, like, could you identify the first ape, or the definitive ancestor of all apes, you won't be able to identify that. That's impossible to
Because
know.
on the evolutionary
So
model,
a better
that's
question
not
to
how it
ask would be,
works.
how do we identify the descendants of a kind, or the descendants of an original design? So that's a better question as to how do we identify archetypes or kinds, and how do we distinguish them from the organisms closest to them? That's a better question.
Right. Sure. Right. How do you define each, or how would you identify each kind? Right. How do you determine that cats are a kind, or are cats part of a larger kind? That sort of stuff.
I actually have a good question that relates to that. A really good question, actually, and that's: do mules produce their own kind?
Oh, I'm not a creationist, but oh, sorry. Go ahead. Sorry.
I would presume those questions are directed at me.
Hold on.
And I'll just say I don't have a good answer.
You don't have a good answer.
If that's fair enough. I mean, I'll tell you when I don't have a good answer.
Are you talking about a good answer
know.
in terms of mules?
We're talking about kinds and both mules, cats, and other kinds. But the discussion is, if a eukaryote cannot evolve from a prokaryote or prokaryote life type creature, isn't evolution falsified, at least at that level? There's no universal ancestry there unless you need miracles. What does that have to do with, I mean, if a eukaryote needed a miracle to make it out of a prokaryote, either we're saying that evolution needed a miracle, or there was special creation. In either case, there has to be a miracle. Maybe there's an explanation. I've not seen anyone, and neither do I expect one. So that makes the claim of universal common ancestry from at least the eukaryotic level very tentative. It is not a foregone fact. And what you've done is you've invoked philosophy. God wouldn't do it that way. Therefore,
no one did that.
Evolution's true. That's a terrible argument.
No one did that.
No one did that.
No one did that. No one did that.
I've made a sign pointed out. Don't explain the eukaryotic nuclear envelope nor the nuclear complexes.
Okay, so a couple of things.
That's a scientific argument. God wouldn't do it that way, or
that's not what our argument was.
Do it that way.
If you took that away, then you misunderstood the entire argument. The argument is not, we are not assuming the psychology of God. We are saying the nested hierarchical structure of organisms is a predicted feature only under evolution. Could God have done it that way? Sure. It's a completely ad hoc and irrational response, but sure, he could have done that. But also, I just want to point out what you did here, Sal. You took something that I don't think any of us are protein experts here. I'm not a protein expert. I don't think anyone else here is.
We need a mutable.
We need exactly. We need someone who is a protein expert on this. But what you're doing is you're asserting that because we here are not protein experts, therefore evolution is falsified. You, on the other hand, said you don't know how to differentiate kinds. Therefore, I should be just as justified in saying creationism is dead. Okay, just because we don't know something doesn't mean that science doesn't exist or that nobody anywhere knows about it. It means that the people you're talking to here on YouTube don't know about it. That's all I'm saying. Does that make sense, Sal?
I think it's fair to say.
On what part?
I don't agree with you, but I think it's fair to say that just because evolution said it's a prediction that there'll be a nested hierarchy, and by the way, there are some subtle problems with that, doesn't mean that it's correct just because there's a nested hierarchy. I gave you a scientific problem that evolution should say that this should be feasible, right? It should be shouldn't, if you're going to invoke a naturalistic theory, it should be mechanistically feasible. I gave you a scientific reason why it's not mechanistically feasible by any natural route. I'm not going to say supernatural, but I'm just going to say it's going to be highly improbable. At some point, something so improbable, one might call it a miracle. But how does saying that evolution predicts a nested hierarchy, therefore it's highly probable that the eukaryotic nucleus should evolve? That's a non sequitur. That's not science. That's faith. That's faith and non sequitur circular reasoning. That is not science. You can believe it.
Okay,
that's
so
fine. You can't explain it, but you believe it.
As I am
that is not.
Again, as I am not a protein expert. Again, I personally am not aware. I'm not familiar with the literature on how this evolved. There is very good evidence that all organisms share common ancestry, and we can trace this backwards through all of the different organisms. We can go as far as you want, and that was part of what they were asking earlier. How do you determine what is a kind? Because when we compare these different organisms, when we compare cats and their close relatives like mongooses and meerkats, we get a pattern, right? We compare their genes, and we get a nested hierarchy. And we can keep on going out like that. The next closest relative, or so mongooses, civets, binturongs, those are all members of Feliformia, right? We can continue the exact same process with the next closest group, which is Caeniformia. Do you disagree that we can do that? Or do you agree that we can do that?
Yeah, real quick, I want to lay down the rules. Real quick, everyone.
I agree.
I want to lay down the rules real quick for my channel and for the sidebar chat. I have zero respect for anti-vaxxers and zero respect for people who spew deadly disinformation on deadly diseases. That is not going to be allowed on my channel anywhere. So if you are spewing deadly disinformation, consider yourself banned. That being said, LBP is here, and I want to give him an opportunity to chat since Sal is not the only, so Sal won't be the only creationist here.
Really, really quickly, can I address something that Sal said a few minutes ago?
No, I'm only going to be here for like 30 seconds. So,
okay,
Dave,
go ahead.
I have not spewed disinformation. I'm sorry if you and Dr. Dan can't comprehend the point that was being made. I have not one time suggested that people are not dying from COVID. Actually, my parents are in a demographic that are at high risk. My brother-in-law has renal failure. I have a sister with who's pre-diabetic, who's also at risk. I am very, very aware of the deadly aspects of COVID. The entire point that was being made is that there's demographics that are at risk and there's demographics that are not at risk. That was the only point that was being made in this whole deal, which has then been somehow translated into I'm an anti-vaxxer and a COVID truther, whatever the hell that means.
By the way,
I don't know what that is.
I never said.
How about you retract your accusations towards me? I'll leave, and you can ban me all you want. And we can never have future conversations. I've always been extremely nice to you.
Yeah, and I've
attempted to have reasonable dialogue. So if you're going to act like I'm somehow being some person wearing a tinfoil hat or freaking alien ears, whatever Speed was just wearing, when that is not anywhere close to the case. And I've had public discourse with members of the Nashville Board of Health about this whole thing on multiple occasions, and a plethora of folks in looking at creating more innovative ways to help people get back to some semblance of normal while simultaneously protecting people who are at risk. So these whole suppositions that are being put forth by you personally and Dr. Dan Stern Cardinal, you can retract them or you can shove them up your ass. I don't care. But that is not how I operate in any way, shape, form, or fashion.
Okay. Well, first of all, I do want to make a public apology to you if you feel that I was misunderstanding what you were saying. I was not calling you an anti-vaxxer. I was just laying down the rules that those type of people are not welcome on my channel, period. But what we were doing is how I interpret it and how other people were obviously interpreting it. Dr. Dan wants a link, so
he was minimizing.
Yeah, minimizing the downplay of it. So that is pretty much the same thing. And that is the rule. Viruses kill by causing these symptoms. Viruses far and away kill people with comorbidities.
And without.
Yes, and without. So he's just showing how ignorant he is of basic public health and virology.
He stated that there are people with no risk for this, which is just patently false. There are differing levels of risk, but everybody's at risk. Yes, people with comorbidities, depending on the particular comorbidity, are at a greater risk than somebody who's healthy. But somebody who's healthy, as we have seen with thousands upon thousands of people in the United States, are still at risk of dying from this. So,
and they have died.
Exactly.
Perfectly healthy young people.
Exactly. So, but there was something. Actually, no. I'll wait till everybody said their word on that particularly, and then I'll go into addressing something that Sal said several minutes ago, if I can.
And I need to get back to something Sal said several minutes ago at some point. So let me know when I have a turn. So.
Yeah, and by the way, LPP, the fact that you wanted to do a public debate in person rather than a debate on YouTube where it is supposed to be free and where nobody is being put at risk says a lot about you. I'm sorry. It does. So we're going to call you Dodgeball LPP, Dodgeball John.
Dodge John.
Dodgeball John, the door is open. Welcome.
He can come back in any time and even out the ground for Sal. I was hoping he'd stay. I guess.
Yeah, I was hoping he and Dr. Dan would be able to join. But,
well, Jackson's here, so I'm not surprised he didn't stay. I mean, if I'm being honest.
I've never actually spoken. This is the only time I believe I've ever been in a room with John.
Yeah, you and him would have some fun. You might have more fun than him, but
I mean, I don't do a lot of debates. I don't do a lot of hangouts, really.
SFT is free to join as well. Come on in, bud.
Yeah, I see you standing for truth. Come on in and join.
I know for a fact he won't join because Jackson's here. So, David, just let me know when I'm free to talk about what I wanted to address to Sal.
I was kind of curious. I mean, I do absolutely want to broach all of these topics. But Sal, kind of getting back to what we were discussing. So, in your mind, do you think that cats are related to hyenas? That the house cat, the tiger, and the lion are all related to hyenas? To you?
Yes. By the way, yes. Angelo, you're welcome to come in.
I don't have a strong opinion.
Yes, our protein expert is here. Welcome, Immutable. Sorry,
Sal, I didn't hear it.
I want to have a protein expert. So he can fill you in why nested.
Say what? I'm sorry. Say that again.
So you agree or not? That tigers, house cats, and tigers are related to hyenas. Do you agree with that? Yes or no?
I have no opinion.
Why?
I doubt it.
Why do you doubt it? They share a lot of genes.
But that's not. I don't. I'm sorry. How does this explain the nuclear?
So you're stuck on this one thing. As I told you already.
Okay, so I wanted to say I respect the fact that you're not a protein guy. I wanted to say I respect the fact you're not a protein guy. I've given you a data point perhaps you didn't consider before, and you've been accepting that eukaryotes can naturally evolve. And maybe I've just put that on the table.
So first off, Sal asked the question, and that was, how do I get a photo instead of an S on my icon? You'll notice at the bottom left of your screen, you'll see a window of yourself there, and it says you're in the show. Everybody can see and hear you there. There should be 3 dots in a vertical manner. You click on those dots. You go to Edit Audio Avatar, and then it allows you to upload. If you hover over the plus icon, it says upload avatar, and you're allowed to upload a picture from your device. But as it relates to the nuclear pore complex, if I may, I would like to read this from an article published in the NCBI, which is due to National Institutes of Health. And this was by Mark C. Field and Michael P. Rout in 2019. And they state, give me a second to re-find it here, that this might be, it won't be super long, but about 30 seconds or a minute. They say, however, we suggest an alternative third model, both plausible and supported by recent data from NPC, or nuclear pore complex structures, where both mitochondrial enslavement and elaboration of endomembrane system occurred roughly in parallel during the transition from archaea to LECA, L-E-C-A, which I believe is last eukaryotic common ancestor. Somebody correct me if I'm wrong on that.
You're right.
This process started with a single protocodomer to begin the formation of endomembranes, which duplicated and diverged into the ancestors of the type 1 and type 2 families of coding complexes in order to help drive the diversification of the endomembrane system into the ancestors of the Golgi, the ER, or the endoplasmic reticulum, and endosomes. Members of the type 1 and type 2 families came together in a partnership to form the first proto-NPCs in differentiating NE. And that's the letters NE. Our model implies that the nucleus, the organelle defining the name eukaryote with nucleus, was actually a significantly later addition on the path to evolution of the early eukaryotes after the formation of differentiated endomembranes and coding systems. This should also be placed in the context of other events that could have taken place concurrently. For example, transfer of genes from mitochondrial and endosymbiont to the nucleus in the formation of cytoskeleton. In a very real sense, this questions the concept of FICA as a fixed point on the pathway of eukaryogenesis. And they continue on in their explanation, and I can post the link to that article in the side chat. Just give me a second here.
Hey,
and
Leo.
like I said, this is from 2019, so this is fairly recent.
Guys, I think I tried to share. What is the name of the paper? I have a share screen, David, that you haven't shown.
I put the link in it. The name of the paper is Pore Timing: The Evolutionary Origins of the Nucleus and Nuclear Pore Complex.
Look at your screen.
Oh, wow. Did you have that up before I said that?
I had that up for a long time. David hasn't shared it yet.
Wow.
So anyway.
Get out of my fucking head, Speed.
Yeah. Anyway, Cell, all you got to do is, all I typed in was how did the nuclear pore evolve, and I got just a ton of papers, and all of them are very good. And you can't just shut down when you run into this. Anytime you guys, and I'm using you guys in quotes, ask how did something evolve, just Google it, and you'll find paper after paper after paper with suggestions about how it evolved. And I've kind of looked into a little bit of the nucleus and all the different ways that it evolved. But the interesting thing is that you come up with like something that is a problem. Are you still with us, Sal? I think he is. Anyway, you come up with something that is a problem. Okay. And you basically you stop right there. You come to a dead stop. Well, I mean, just because we don't know, and nobody may know yet how some of this stuff evolved. We are at the very early stages of this. That does not mean that it could not have happened just because it's a problem. And you do that over and over again. You come up with this dead stop. This is just too improbable. We're going to throw up our hands. It couldn't have happened. So that's what I wanted to say.
It being unexplained does not mean the explanation is not natural. That is, by textbook definition, fallacious.
Which is exactly what I was saying earlier.
The abundance of papers that have technical flaws in them, and I could see it right there, is not evidence that this problem is solved.
What particular error?
I don't think anybody is saying that it's solved, just that there's potential pathways for how it could be solved.
I pointed out something with localization signals. For whatever proteins there are that are shared between the prokaryotes and the eukaryotes, assuming there is a transition, I pointed out there have to be 2 sets of localization things put on every protein, that when we have a eukaryotic nucleus, you're going to have to reformat all the proteins. That's a localization problem.
But we don't
They
have
just
to
totally
reformat all
ignored
of them.
that.
We share the chaperonins with archaeans.
And how many prokaryotic cells do we see having endomembranes in them with nuclear pore complexes that are just floating around?
Why is it?
That's a totally, that's an assumption they made up. It can't be tested. That paper has something that can't be tested, can't be experimentally verified. They just make it up and represent it as a solution.
Why is it not
that is not
possible
science.
for these proteins to be transported by vesicles? As many proteins are when they're transported via endocytosis, typically. That appears to be like.
I believe that's
The
what RJ calls
proteins
a
have to
deer in the
be-
headlights moment.
So we have 2 things.
Go ahead.
We have, okay, so there may be some vesicle transport, but the reason, there's definitely vesicle
Well, there's
transport
vesicle
across the
transport,
cells.
not only. Hold on.
But the reason that it has to be, things don't transport across the nucleus. You have the nuclear pore complexes there. Stuff is transported through there, and it's regulated. You just don't have anything going through there. It has to be regulated what goes in and out. And it's not just the proteins, it has to be the RNAs too. And that's why there are localization signals that tag the proteins
right, okay.
where they have to go.
I don't see an issue why you.
Aren't there localization
Sure, and
signals? Aren't there localization signals in the proteins?
Are you talking about?
Okay, the vesicles don't use. Do the vesicles use localization signals to locate where
I'm confused
the
if you're
protein's going to
talking
go?
about like interprotein folding, like when proteins enter, for instance, when proteins adopt a structure based on going into, I forget the precise name of the proteins, but chaperonin proteins essentially that allow the protein to fold, or if you're referring to the process of transport folding essentially, where proteins fold while they're being transported across a membrane. And those are 2 entirely different mechanisms. But to answer your question, why is it not possible to have a protein such as, say, a fibril molecule, or an actin filament, which does transport molecules from one side of the cell to another, and the receptor at point of that molecule happens to be a molecule that is commonly associated with the nucleus?
It's possible,
right?
it can be transported.
This idea that you need proteins all the time, not really. A lot of things in the cell, even for like extracellular transport for proteins that get exported and are expressed outside the cell, have that mechanism. So I don't see why you always need new proteins necessarily. Why can't it be an adaptation?
Well,
why can't it be an adaptation of existing molecules, and just that the paths that they're, or that the locations of cells that they're targeting is different?
Well, let me give you an example then. Let's say we have a prokaryote and the genome is now located inside a nuclear membrane. What's the probability that you'll get the right proteins in the right amounts being exported, or let's say the RNAs
being exported
in the nucleus and then going to the ribosome? What's going to happen if the creature's dead?
What exactly do you mean by export? Eukaryotes don't export their genetic material. Prokaryotes do, because prokaryotes have plasmids that essentially serve as a genetic unit. Okay, but what do you mean by export? Are you talking about are you talking about the way that RNAs are transcribed as pseudogenetic material and then used as a transcript for protein translation, or what?
I'm talking about.
Go ahead.
Oh, I'm sorry.
No,
I didn't mean to cut you off.
I'm confused by what you mean by
delayed.
transfer of genetic material. Because typically when we're talking about transfer of genetic material, usually it's referring to processes like horizontal gene transfer or something like that, which is a strictly bacterial phenomenon. It's not one that occurs in eukaryotes.
All right. All right. I see we're talking past each other. I was talking about the nuclear pore complex, and I have a diagram here. I was talking about the nuclear pore complex.
Right.
So you have a nucleus, and you can transport RNAs and proteins across that membrane. We have things called importins and exportins to facilitate this.
Yeah.
Is that correct? In fact, I have a diagram here of the one of the ribosomal proteins, RPS20, that needs an exportin. So this is really incredible. The RNA that codes for it has to go through the nuclear pore complex. It originates in the nucleus, goes out through an exportin, presumably. It then goes to a ribosome. It is then translated, and then it is imported back into the nucleus by an importin, goes through more processing, and then it is exported across the membrane. That sort of orchestration is not reasonable from using vesicles. It has to be timed, and it's especially important because this is a ribosomal protein. It forms part of the 40S subunit. That's what I was talking about. So there is a video on nuclear import-export, and that would be valuable to anyone discussing this.
I gotta ask a question.
The problem
here.
I have is how can they make these assertions?
Okay, but so the question I'm asking in particular in regards to that is that I'm saying not only just vesicles, but vesicles that are responsive towards certain receptors, right? I understand the process that it's being viewed, that it's undergoing endocytosis and exocytosis in different intracellular regions, but I don't understand, is that process uniquely different from cells using passive or active diffusion normally to transfer material that normally wouldn't be permeable to?
This is different.
So it's a mechanism that is not...
This is a different process. With vesicle transports, we're having the protein or the whatever is enclosed by lipids. In this case, the thing that enables it to go through the nuclear pore complex are other proteins like importins or exportins. So it's a different system. Now, the thing is one could invoke vesicle transports, but how's it going to be orchestrated? For example, this RPS protein, I have a diagram here, one can look it up. I'm sorry, I don't have the citation immediately. I'm not sharing my screen because it's going to shut everything down.
Okay, but so I can answer that question for you.
Can I ask you a really quick question, Immutable? Really quick.
I just want to propose a question to Sal first. Is it not possible for the... Hold on.
Yeah. So
is it not possible for, say, a serine or a threonine residue on an amino acid to form an ether bond with the lipid portion of a molecule to become covalently linked to that lipid?
Well, if you say it does, I'll accept
I'm just trying to say, are there post-translational modifications that can occur in such a way that there can be a temporary linkage between a lipid molecule and an amino acid? Because certain post-translational modifications are responsible, for instance. Okay, but I'm not saying...
I'm not aware of it. I'll accept it's true.
I need to ask a question.
I'm fine with that. But to effect a post-translational modification, you need a sequence that'll be recognized properly by, like if it's a serine, are you talking about a phosphorylation to enable this? Then you would need a kinase to do that. The kinase has to recognize the right location on the protein to enable this. So if you want to invoke post-translational modifications, you're adding actually more complexity. But I mean, I totally respect you, Immutable, and I appreciate you joining. This is a better discussion now. because
To-
we're
to
actually
my
talking
knowledge,
about the mechanisms. Leo, I'm sorry
I mean,
about
to
that.
my
I really
didn't
want to
really-
ask Immutable a question, just
Sure.
so that I ensure that I understand. A nuclear port complex, isn't that just like a complex of proteins in the nucleus, or is that too simplified?
It's been a hot minute since I have actually
It's
looked.
like it facilitates the movement of molecules into and out of the nucleus.
Okay.
It's a permeable
That
barrier
makes
between
sense.
the nuclear.
Okay,
between
can
the
I-
nuclear-
Can I just read a little quote
Nuclear
here? It's
port
quick.
No, I'm actually
The
kind of
nuclear
interested in
port
this conversation with Sal so far. And
Yeah.
correct me if I'm wrong, because you would probably know about this more than I do, Sal. But to the best of my knowledge, post-translational modifications do occur to some extent in prokaryotic organisms. And for the most part, a lot of modifications made to the enzymes usually have a recognition sequence of about 3 amino acids, if I'm not mistaken.
Well, the post-translational modifications in prokaryotes is not on serines and threonines and the aromatic groups. It's on histidines and others. That's also another problem
That's
for
a-
evolutionary
that's a
biology. Why did the kinases
I mean,
and phosphatases start being oriented toward different amino acids in prokaryotes
I can
versus
list several
eukaryotes?
different amino acid modifications that occur naturally within cells. And lipidation and the formation of ether bonds with serine and threonine is one of them. But so, I mean, I don't understand why it would be an issue for post-translational modifications to occur if what post-translational modifications are already is essentially catalyzing a reaction between two molecules that's already favorable.
I'm willing to accept it as for the sake of argument, and that's an interesting point. The point that I'm pointing out is an example, this RPS3, which is part of the 40S ribosomal subunit. This is a really critical protein. It may be, let's just suppose it's a necessary condition that we have this lipid transport. It is not a sufficient condition to enable it to move in this orchestrated way where we have the RNA go to the go out of the nucleus. So the RNA is coming out of the nucleus. It then gets translated by the ribosome. The ribosome then releases it. This RPS3 goes back to the nucleus. Then once it's in the nucleus, it's going to undergo more processing. Then it goes out as part of the pre-40S unit through the nuclear pore complex. That's multiple steps that have to be right because this is such a critical protein. The creature will die because ribosomes are important for cell life. And so even though you've indicated some very interesting chemistry, it's not a sufficient condition to do this orchestration. And why have something so complex when the prokaryote just works so nicely without all this
Why?
weird Goldberg complexity? And that was my point. By the way, that ether bond, that was pretty sharp of you.
Yeah, I mean,
wanna
to
compliment
give
you
a
on
basic
that.
answer to your question, most proteins that are expressed in prokaryotic cells, if I'm not mistaken, tend to undergo native folding. They don't tend to undergo folding in the same sense that proteins that eukaryotes express. Eukaryotic proteins tend to express tend to be expressed and then folded by chaperone proteins. Whereas most proteins in prokaryotic cells are more or less flexible in that they fold based upon the environment they're found in. Having an ability for a protein to essentially be stabilized by other residues and for it to fold in an area where it's secluded from other members allows for more computation. Well, it allows for a broader span of biochemical reactivity and diversity.
Shut it a little bit, just so the sound doesn't go up the staircase.
Could I interject for a minute?
Go ahead.
But I found another paper that's basically similar or to the point. Extensive reduction of nuclear pore complexes in nucleomorphs. So they found like remnants or basically real simple archaic forms of nucleopores, and they found some nuclei some nuclei without pores at all. in some organisms. I believe some bacteria. I'm not sure if it's archaea or whatever. But anyway, so the thing is, you said they haven't solved the problem. None of these papers are saying that they solved a problem. And I think you're projecting what you're doing, and what you're doing is you're trying to prove that it's impossible. And you're saying they haven't solved the problem, so it's impossible. All of these papers have one little tiny bit that they add to the complex, to the problem, to what we're trying to learn. None of these guys have ever claimed that they solved the problem of how nuclear pores evolved. They're working on it. And each little paper, and you're going to have 1,000 papers on that subject before you're able to put together the pathway that it's solved. But your problem is that you have to prove that it's impossible. And you're proving that it's impossible by noting what we currently have today. The very complex nuclear complex that is 3 billion years down the road. And I just think that is just a invalid argument.
I see that a lot, that, well, when life first developed, it had to look exactly as it does today, when literally no reputed origins of life researcher or biochemist would make that statement. They would argue that it looked significantly more simple and more chaotic and slowly evolved into what we see today, which those, and I'm sure Immutable can attest to this because he has a degree in biochemistry, if I'm not mistaken, that how we think of various cellular processes and genetic processes, it's actually a lot more chaotic than all those fancy schmancy animations would have you believe. It's significantly more chaotic than that.
Okay,
Yeah,
I would like just to quickly read here from my site. Each nuclear pore complex works so fast it can authenticate somewhere between 520 and 1,000 pieces of cargo per second. A typical nucleus has about 2,000 to 4,000 or more of these gates, which are made up of 30 or more very complex proteins. They all have to be disassembled and reassembled every time a cell divides. I think this is staggering. If you imagine that it can recognize between 500 and 1,000 pieces of cargo per second. And that is fantastic technology.
Do you know how many gluons are exchanged between a quark and in a proton per second?
That has nothing to do with protons.
Well, it's called an analogy. You're arguing that because this happens really, really fast, therefore it's not natural.
I've not said it's not natural.
Not a valid
I've not said it's not natural, Leo. I've just pointed out about the complexity and technology.
Do you know how complex strong interactions
the nuclear pore? And of course, I mean, in order for the nuclear pore to recognize each molecule, if it can go inside of the nucleus or not, it requires very sophisticated recognition mechanisms, and I don't think science actually even knows
how does a gluon
works,
recognize the color of a quark to change its color
Can I finish, please?
If you're not long-winded, yes, awesome.
Wow.
But there are 1, 2, 3, 4, 5, 6 other people in the room.
Yes, but Saul and Immutable, they have talked for 10, 15 minutes. We have
because
just
they were having a back and forth.
I tried to make a point for 2 or 3 minutes, and you are simply again interrupting me. And you know that I don't like that. So please respect
your own argument here.
Okay, so. Again, Leo, I think you should learn to respect when someone is talking.
I do. It's just,
I hate it when
gets annoying hearing literally the same things over and over again.
I have the right, if I'm in a chat with other people, to make my point without you interrupting me and saying that I am annoying. You are annoying as well, but I am respecting when you are talking. I acknowledge that. And I am not interrupting when you are talking. So please stop when I am talking to you.
Let me reiterate my question.
No, I mean, I have made my point. I
recognize
I am not finished
quark
yet, Leo.
when it is exchanged between them. There are things significantly more complex than biology in our reality, and we've explained them. This idea, well, this is super complex and you can't explain it, so therefore it was God magic, is the dumbest fucking argument I've ever heard in my life. You are literally appealing to fucking magic and then claiming that's more reliable than testable, repeatable, natural explanations. It is the most disingenuous and dishonest thing I have ever fucking heard. And it gets tiring hearing it over and over and over again. I'm done.
Okay.
It is aggravating.
Yeah.
But
Yeah, on Tanslo, you're always welcome here, but it does get annoying when you bring up the same points every single time, and we try and start another conversation while we're in the middle of another. So, Immutable, I'm sorry.
No, I was actually enjoying the dialogue me and Sal were having up until Tangelo decided to do a gosh gallop. So
That's the thing. He bitches that people interrupt him, but if you don't, he'll go on like Nephilim for 10 fucking minutes. Nobody wants to sit here and listen to that. We want to take it point by point. You said, well, this recognizes that. Okay, cool. Let me give an analogy that demonstrates why you're saying what you're saying is bullshit. How does a gluon recognize the color of a quark to change the color of that quark when it's exchanged between quarks? Except that it doesn't recognize it. It's just fucking physics. It's just shit that happens. It's not it's this anthropomorphization of reality that they don't understand, that these are just simplified explanations of extraordinarily complex things. They take the simplifications literally and then try to make arguments from it. It's just dumb. I'm sorry, but it is.
So I hope if nothing else that we can agree on, it's that cells and the biochemical pathways that are contained therein is a very chaotic type of process, if you will.
I really want to make another point. Notice how
yes and
Sal
no.
isn't quoting from his own blog. He's making his own arguments. I might not agree with them, but he is standing on his own 2 fucking feet, not consistently relying on quotes from his own blog to make his points. And that is the defining difference.
Yes, I agree 100%, Leo.
yes.
But Sal, back to the question.
Yeah, sorry.
Yeah.
Go ahead.
It's actually an interesting problem in biophysics. So how something so chaotic can also lead to organized outcomes. Just for what it's worth, I was very interested in transport. How, like, say, a link RNA transcript will emerge from chromosome 12 and find its way to the right location in chromosome 2, the Hox cluster. It doesn't have any active propulsion. It uses, for lack of a better word, Brownian motion. That's not exactly the right term. It's kind of close enough. So yes, it's using the chaotic motion, but it's able to do it in a way that it can still result in navigating to precise points. So when you asked, is the cell very chaotic? Yes, in terms of the molecular motions, but there seems to be something in the way that it's organized and the way the chemicals are positioned. It's able to do it in a way that kind of mitigates all the noise, like a sailing ship can actually utilize almost random air currents, but then it's able to navigate to the right place, which is an interesting problem in biophysics. That's
I suppose
like
I was more
biophysics.
referring to the fact that, like, it's a useful thing that cells happen to settle on this concept of metabolic convergence, that being that when metabolism occurs, it tends to generate usually a list of about 10 to 12 chemicals that are identical in structure, and those substrates can be further transformed or retransformed into whatever they're needed for by the cell. But because you have this phenomenon of universal convergence or metabolic convergence within cells, sometimes interesting things happen with cells where you have an enzyme that tries to catalyze a reaction, but it gets blocked for reasons like there's something else that can bind with that enzyme in an allosteric position, and it just makes it stop for the time being. Where what I'm talking about is kind of, even though a lot of metabolic cycles are controlled, there are very interesting occasions where it's kind of messy for them to be managed.
I'm sorry, I gotta chime in. Sal's chat is all about, oh, Leo's triggered. God, these people are so pathetic. Oh.
Well, I'm glad we could provide some entertainment for you tonight,
Oh,
Leo,
good.
and put a smile on your
Redefined
face.
living is just straight going off.
So,
Sure.
Immutable, I just had this question. I think I heard you say you believe in metabolism first, or you gravitate
I would
toward it. I'm not going to.
I would gravitate towards it personally because I think that, I'll put it this way, I think that there's an appeal towards metabolism first in that if you have systems which are closed, in the sense that if you deal like with a lipid system, for instance, where you try to argue that very basic peptides are forming and binding with RNAs or whatever the case may be, the issue I have, and it might be my naivety just speaking, is that it seems that simply relying on the free energy that's available to that type of kind of closed system doesn't quite seem sufficient to be useful in terms of providing enough energy for the cell to complexify sufficiently. So I could accept like that in an environment where a protocell could form, or like conventional hospital models or things like that. You might have amino acids that form of like, say, 10 to 15 in length, and that's being really generous. The issue is that those proteins, or they're not really proteins, really, they're peptides, actually. These peptides are very short, and it seems that they would adopt very limited structures and that their catalytic activity would be limited as well. Whereas if you have a metabolic system going first, especially one that's autocatalytic, it seems that that type of system would be more supporting or more allowing, I guess, if you want to put it that way, for peptide diversity to grow and exponentially.
The question I just as a follow-up question, in the metabolism first model, do you hypothesize the genome exists to be able to have recipes
Define
for
genome?
To create enzymes?
That's the major thing, and it's the reason I don't really fully endorse metabolism first, is that you could argue, for instance, there's been some papers that have argued, for instance, that the Krebs and citric acid cycle could be an example of a system that was occurring at the origin of life. That this basic kind of cycle of converting between, I believe it's citrate and the various chemical reactions it goes through, could have been possible. The issue that seems to be encountered with metabolism first is that, okay, so you can have proteins that form, or peptides, I should say. I keep forgetting that. You can have peptides that form, and you do have nucleotides around, no doubt. But the only inference you can really draw is the relationship between protein and metabolism, not between the amino acids of that protein and the genetic code. And that's something that you have to account for at some point. You have to explain in some model for the origin of life how and why it is that this pair of 3 codons happens to specify for hydrophobic amino acid like glycine or alanine or something like that.
Did you ever get the paper that I sent you, Immutable, on how the Krebs cycle could have actually evolved?
Yeah, I did.
Okay.
I need to reread it again, but I do think I have that in my folder.
Okay. It's from Nature Journal, so it's
There we go.
I believe it's from, let me double check. I believe it's from this year. Yep, 2020. Yep.
Nice. But yeah, so Sal, to answer your question more succinctly, I know I'm rambling.
No, I was actually curious. That's fine. If anyone that I'd love
Thanks.
listening to rambling
Appreciate
into
it.
you.
Yeah, metabolism first.
I was just very, very curious because I put that on the table today. I was talking about the topoisomerase, and there are other enzymes that need a genome to be able to replicate them, and definitely the citric acid cycle. I just have it here. I used to know this by memory, by the way. I no longer do. I actually had to recite all the steps. So let's see, there's the citrate synthase, the acatinase, isocitrate dehydrogenase, alpha ketoregulate dehydrogenase, succinyl CoA synthase, succinic dehydrogenase, fumarase, malate dehydrogenase, and then there's carboxylase and pyruvate dehydrogenase. Those are several proteins that it's like, okay, let's say hypothetically they emerged. It'd be pretty desirable or necessary that we need to create more of these for the next cell. And that's why I think it needs some sort of genome.
That could be the case, although I could also see it being the case where early systems rely on a type of more inorganic-based reaction. So where you have this kind of reaction being coordinated by a metal. But later on, it becomes the case that amino acids with residues that coordinate well with this particular atom are able to catalyze the reaction quicker, because obviously now you have an ionic compound or covalent compound that's more stable, and thus when it gets into I guess when it comes into contact with the appropriate substrates, it's able to catalyze that reaction on a more consistent basis. Although that's purely speculation. I have no way of knowing if that's actually true or even plausibly correct.
Well, thank you. You gave me pretty good responses there. I was just curious, and I was throwing out why. Obviously, being a creationist, I think the everything first model makes the most sense to me, which is, when we say everything first, that's like special creation. I don't think that the cell, there are enough parts that have to be there simultaneously.
What I would ask, though, what I would ask though in this case is more so the question, and it does lead to abiogenesis in an indirect way, but is to ask the question, and it's still very much so a open question in abiogenesis research, actually. But what is the minimal criteria for a eukaryotic cell to function, essentially? Because one of the things that should be noted is that, and it should be expected, actually, that eukaryotic cells that were even first emerging probably do not have all the functions that modern eukaryotic cells do. And just as one basic example of that, take, for instance, plant cells. Plant cells don't have the ability to separate their cell walls from each other. When a plant cell divides, essentially it shares the same cell wall as the parent cell.
I think it should also be pointed out that there are organisms alive today that don't have all of the quote normal cell parts. Like tapeworms don't have peroxisomes, and there are certain protists, and even recently there was a nidarian that was discovered that doesn't have mitochondria.
I was going to ask you, David, if you wanted me to throw the link to Matt, because I like Matt and I wanted him to come in, but you already put the link in the chat. You beat me to it. Welcome to Speed, by the way. Go ahead, Jack. I'm sorry.
Oh no, I mean
Yeah.
that was it.
By
I
the
was
way,
just saying.
real quick, we are wrapping things up, but I want to start wrapping things up because I'm falling asleep. Matt and I, and others, I am hosting an open mic again tomorrow at 9 p.m. on my channel Eastern Time. So please be here at 9 p.m. Eastern Time tomorrow, because it'll be hopefully a fun topic. And just have a fun open mic. So yeah, these open mics are becoming more popular by the day.
Hey, there we go.
Can I ask? I need to ask Immutable another question.
Oh,
On
go
a
ahead.
scale of 1 to 10, how versed are you in special relativity?
About like 2.
Okay,
The,
never
the
mind.
main thing is like I can conceptually understand it fairly well.
Yeah, which I don't think it's hard to, but yeah. You know.
I'm trying to like multitask, learn like special relativity, linear algebra, and like some basic abstract algebra right now. That's kind of
Course load.
I won't lie to you. Abstract algebra is probably the hardest thing I've seen, and I've seen some like Chinese algebra problems, but this takes it to a new level. Good God.
I will say, if you can get linear algebra, I'm not going to say abstract algebra won't be hard, but it will be a bit easier than it otherwise would be because
Yeah, I'm trying to
it's a
understand.
lot of matrix mechanics in both. So
It's easier than calculus.
The thing is,
no.
the
What?
thing is,
I thought calculus was a breeze.
oh yeah, that's what everybody describes about calculus. It's just so fucking easy that everybody gets through it so nicely.
Right.
Although Calc 2 kicked my butt. That Calc 2 was hard.
You get to like tensor calculus, and then you're like, nah, see, this is a little different.
You know, Jackson, what's actually funny is that I did much better. I think I got an A in Calc 2, but I got like a C in Calc 1 for whatever reason.
Okay.
Like I don't know why. I think part of it was because like once Calculus 1 bent me over, I was like, all right, I'm kind of getting the hang of this integration thing.
Okay, so here's the thing with me. I did not. I like skipped the class Calc 1, or I mean, I skipped going to the class, but I passed it. I went to like all the tests and just ended fine because I would like read the book at home, whatnot. I just thought it was a breeze. But I went to every single class for Calc 2, and I was still struggling with the math. I was like, oh my god.
You see, that's the weird thing. Like I went to every class for Calc 1 and 2. Calc 1, I just could not get my head around.
Do they introduce partials in Calc 1 or Calc 2? I don't know because I've
Calc
never been.
3.
It's Calc 3?
Yes, the reason for that is you only need to introduce the notion of vectors and partial derivatives because when you define three-dimensional functions, it's important to define unit vectors, and that's also convenient to define things like dot product and cross product.
Yeah.
Because typically you're taking, like if you're taking like a line integral or something like that, you're defining it as, you know, the integral over the boundary of an object, or, you know, the divergence or curl or whatever.
Okay, so del is involved. The del, like del dot and del cross, are involved. Yeah.
That's essentially the manipulation of fairly basic matrix mechanics to deal with
Fairly basic, of course.
Well, I mean,
fairly basic
compared to like differential geometry stuff.
Differential geometry. I'm sorry, like that and perturbation theory have kicked my fucking ass, and group theory. But group theory is just God mode. I don't care what anybody says.
Group theory is easy.
Fuck off, Speed.
Supposedly, if you understand like linear algebra, group theory. Well, go ahead. I'm sorry.
For what it's worth, I've studied general and special relativity, and I have some derivations. We could take Maxwell's equations and show the Lorentz transformation. I'm going to be publishing a educational paper, not a peer-reviewed, not a science journal, just educational thing that will show that as calculus exercise and using partial derivatives. And just
the weird thing is. The weird thing is for me, like with general and special relativity, is that the math makes sense, but God, the notation is a nightmare. It's like,
especially in general.
No thanks. I took my math. I am content with my animals. Give me my animals. I'm going over my corner. That's it.
I have the unfortunate curse of actually liking certain branches of mathematics, and so I'm kind of like really interested in certain aspects of it.
Through high school and like into college, well, kind of at first, I was like, okay, math is fine. And then as it got like progressively more difficult, I was kind of like, I don't like this so much, but I do like my animals. I can arrange all my animals. That's all I care about.
The interesting thing is that what reignited my interest, because for a while when I was taking like Calc 1 and Calc 2, I was like, okay, this is kind of pretty cool.
Mm-hmm.
But I think it was after Calc 2, I got introduced to the famous like Euler identity. E to the I pi plus 1 is equal to 0.
Yeah.
And for me, that really like reignited my interest in math, just because seeing that two transcendental numbers could relate to each other like blew my fucking mind. I was like, what the hell is going on? Like, here we have the exponential constant, and here we have something that pertains to circles. I don't understand how these two connect, but I do understand how the formula was derived. Somebody help.
I get lost in general relativity when it starts. It's the one thing I've had a lot of trouble with is the Riemann tensor and curvature and Riemann tensor fields. I'm like, what the fuck? There's so much there, and I look at it and I'm just like, I get half of it. The other half, I'm just like, I don't. What the fuck are you telling me? I don't get it.
I only needed math to do like physics 1, and then I was done. So.
The worst part of general relativity is the stress energy tensor. I still don't totally get that.
I know what it is, but
tensors.
I couldn't tell you really. I
don't think anyone in my class.
It's the energy. It's the stress that energy puts on space-time, hence the term stress energy. But in terms of mathematics, don't ask me anything because I don't know.
That sounds whack.
It is whack. Like I was getting into general relativity. I'm just like that one meme with the black guy in the glasses and he's narrow-eyed and he's just like whack. That was me. I was just
that's
like
Hannibal
whack.
Buress.
Yeah, I was literally just like, I'm reading this book and I'm just like whack.
Can I tell you a story about general relativity class? I'll tell you a story. The professor encouraged us to use Mathematica because the tensors were so huge.
Yes.
And he gave us 2 weeks to solve to show that a particular metric satisfied the Einstein field equations. And even with Mathematica, guys were struggling to get it there. Yeah. So the computers were getting it, doing all the heavy lift, some of the heavy lifting, but setting it up. And these are bright guys in my class, and that's how bad some of this was. And so, I mean, I can imagine before the age of computers, we had professionals just spending an entire year trying to do all the equations, because like the Einstein field equation, some of the metrics have 200 terms. And there a few times I was deriving some things, just part of it. And it was like, oh my goodness, all the partial derivatives and all. Yikes. And the, what did they call it? The chain rule and stating the chain. It was like 20 pages. And I did that by hand, and then someone said, "Oh, use Mathematica, professor." Didn't you hear the professor said to do that?
I have a question now.
But then I at least I got a feel for.
Were the derivations approximate or exact that you guys had to give?
Okay, no. They're exact, but there are only a few things that you can solve exactly. There are only a few solutions to the field equations that are tractable. And one time I was joking with one of my colleagues, my fellow students, I said, I don't think anyone can solve that particular kind of field or shape. I said, I don't even think Einstein would solve it. And she knew I was, there's a element of truth to that these equations may describe general relativity, but the solutions, very few people are able to come up with anything tractable. So they have all these kind of nice, tidy models. And we know they're inexact, but if you try to make them realistic, they'd be a total mess.
I know I believe it's
we only are able to solve.
I believe it's the Friedman, Lemaitre, Robertson, Walker metric is one of the most favored exact solutions to the equation. It's actually a standard model of cosmology for how exact it is, so to speak.
Yes, and that's the easiest. That's the easiest one, and so that's why it's studied. It's one of the easiest ones. There are others.
I can't think of them right off the top of my head, but I do know there's others. Yeah.
Another one is the
there you go,
metric.
Schwarzschild.
That's not too bad for the. Yeah. So I don't know how we got talking about.
Someone said math is fun, and then we just jumped right into it.
All right.
We're chaotic evolution.
All right, guys. I am falling.
Before we go, let's see. I'm going to talk to Otangelo tomorrow night at 7 p.m. Central Standard Time on my channel. And Sal, I want to talk to you about these nuclear pores and the problem at some point. But and also,
thanks.
Leo and I are going to have a debate on modern day debate. Is it on consciousness?
No, standing for truth, not in.
No!
What was it, December?
Did I agree to that?
Yeah, you did. It'll be fine.
Not
moderating. When he's moderating, standing is not that bad.
Oh, but no. Let me rethink this.
No.
I had a discussion with Kevin Anderson on Standing's channel, and he didn't really moderate. I just kind of we would just both kind of stop and be like, "Okay, no, you go. All right, no, you go."
Yeah, and that's the thing. Like when you can moderate yourself, it's good. But it's going to be. You said you needed 2 weeks to prepare, so I think it's
the 14th. The 14th.
Yeah, the 14th of what, January?
Yeah. So, Sal, I have to work.
There's
on that.
Year after 2020.
Apparently,
this is news. This is news to me. Okay.
Holy shit. You guys
the problem isn't 2020. The problem is the 20s. The cursed decade. Has that occurred to anyone?
I don't know. But Sal, we need to have a discussion on relativity at some point.
Yes. I wanted to immutable. I'm gonna kind of redo our talk on quantum mechanics. That was a real good start, and you'll see I practically plagiarized half of what you did. I just wanted to give you a heads up. What I'm going to be doing is just kind of so you're welcome to come to my talk, but it's going to be basically the same one you gave, but then I'm going to add some stuff to it because I found some, because I want to make a teaching module on Schrodinger's equation and the partial differential.
If I can just say something real quick.
So
I don't know if you want to go like head deep into that immediately. I think, and I'll say this personally, I think what's a lot easier is, and maybe you've accounted for this already, it makes more sense to people if you compare like the classical model, if you form the Newton's equations in their differential forms and explain kind of like the discrepancy between what exactly is the differences between those two, because they are actually very similar to each other, and the only reason that the Schrodinger equation has a different solution is because of that negative term that pops up, the negative h bar squared over c.
Yeah, I was gonna say, isn't it like complex numbers and things like that that take into account aspects that Newton didn't obviously know about?
Well, I'm gonna go the safe. Yeah, I'm going the safe route and just kind of sticking close to the textbook, but then I'll add things that help me understand, because in the process of our discussion, I found it helpful to restudy differential equations.
That kind of helps,
solutions.
because especially if you want to go through separation of variables, which is something we didn't quite go through.
We still need to do Brockett. We still got to do Dirac notation someday.
Yeah, luckily Brockett isn't too horrible of a thing.
It's not bad at all. It really isn't.
And the thing is too is that the majority of like the ugly math that shows up in quantum mechanics is mostly dealing like with integrals, especially when they require you to take a Fourier transform, which essentially just, you go to a reference book, open it to a page, and see, okay, I have these two functions multiplying with each other, and the result is, there we go.
All right, guys. Well, we have been going for almost 3 hours now. I am starting to fall asleep. But if you've enjoyed this conversation, there is going to be an open mic tomorrow at 9 p.m. Eastern Standard Time. So I hope you all come back for that. We can definitely continue our discussions then. Just wrapping everything up. Thank you guys so much for coming. We've covered a whole lot of different covers, and this is one of the reasons why I love open discussions, is because you start on one topic and end in a completely different direction. So I'm going to let everyone maybe plug in their channels, give everyone the any last minute things you want to say, starting with Jackson. Any last words, Jackson?
Not really. This was fun. Thanks for having me on.
Yep. Hopefully you guys come back tomorrow. Immutable, anything you want to say before we leave?
No, I mean, I talked to Otangelo yesterday, which was interesting to say the least. I'm just kidding. I enjoyed that conversation. And now I get to talk with Sal, two of my favorite creationists. So, yay. This is a fun conversation. It's always nice to see what people on the other side of the fence think, and for me, I care less about what people believe and more so why they believe what they believe.
Yep.
The actual arguments they use to come to their conclusions are more significant. Because ultimately, even though I have my favorite options on what I think regarding origin of life or whatever else, the universe doesn't really have to oblige to my wishes of it.
Exactly.
And so whatever happens to be correct will be correct, and I have to accept that.
Yep.
This is fun.
All right. Speed, any final words?
Yeah. Let's see. I'm about.
I'm an old fart.
I'm 10 subs away from 200. Subscribe to my channel because I'm going to die soon. I'm really old. And the conversation with Otangelo is kind of the beginning of something. So that's going to be cool. And Cell, look forward to kicking your nuclear pore. So, and that's it.
Leo, any last words?
Yeah, I'm 12 subs away from 200. So subscribe to me before you subscribe to Speed.
I
know.
So.
I gotta beat Leo.
I just put out a video today that talks a little bit about causality. It's about 10 minutes long. I'm thinking maybe this weekend, if not earlier next week, I'm going to do a live stream, a raw live stream on Hunter Braxton's 10 Questions for Atheists. I watched like the first maybe 10 minutes of that, and just to see what it was about. And I figured, you know what, I should do a live stream on this where I haven't seen it before so that my responses are raw, uncut, and genuine. So I'm going to be doing that at some point this week at Leophilius on Twitter, Leophilius on YouTube. So subscribe. I got a lot of content coming. And do subscribe to Speed. He's a good guy, and you should definitely be subscribed to him. If you're not, I don't know what the fuck you're doing with your life.
Yep. And Sal, hopefully you enjoyed the conversation, and I want to thank everyone for having a mature and respectful conversation. Hopefully everyone felt that they were given equal time. And Sal, anything you want to say?
Yes, this was a very good opportunity to kind of test out my computer system, and the simulcast is getting a little better each time. And I'm finding I shouldn't use the computer I'm on to be doing this because it's slowed down. And I'm just really grateful that we were able to continue just from a technical standpoint, because I just about faded away there early on. I also want to thank David and Jackson for filling in while I was taking care of my mother. And I'd like to wish everyone happy holidays.
You too, Sal. Merry Christmas.
And guys, it's been great
happy Hanukkah.
seeing you all again.
Yep, happy Hanukkah, Merry Christmas,
Merry Christmas,
whatever you celebrate.
You know we love you, Sal. You know we love you.
Yep, and I hope your mother does well, and I am thinking about you and thinking about your mother as well, so my thoughts are with you. All right, everyone, that's a wrap for tonight. Join me at 9 p.m. tomorrow for another